There's a place on this planet where the sun has never reached, a place where the pressure would crush steel, where the cold seeps into everything, where silence is not an absence but a presence. It is not on some distant world orbiting a faraway star. It is here. It is beneath us. It is at the bottom of our own ocean, hidden beneath miles of black water, waiting in a darkness so complete that the very concept of light becomes meaningless. The Mariana Trench. Say those words and most people conjure an image of emptiness, a void, a crack in the Earth's crust where nothing could possibly survive, where the laws of biology as we understand them simply cease to apply. For centuries, that is exactly what the scientific world believed. The deep ocean was written off as a dead zone, a sterile wasteland of crushing pressure and freezing temperatures, as inhospitable to life as the vacuum of outer space. It was a convenient assumption, one that allowed humanity to turn its collective gaze upward toward the stars and outward toward distant continents, all while ignoring the greatest unexplored frontier on the planet, the one directly beneath the waves. But that assumption was wrong, spectacularly, profoundly, beautifully wrong. Because the Mariana Trench is not empty, it is not dead, it is not a void. It is an ecosystem, a living, breathing, pulsing community of organisms that have adapted to conditions so extreme, so far beyond the boundaries of what we once considered possible, that their very existence forces us to redefine what it means to be alive. Down there, in pressures exceeding 1,000 atmospheres, in temperatures hovering just above freezing, in a darkness that has persisted unbroken for billions of years, life not only exists, it thrives, it innovates, it endures. This is the story of that life. This is a journey into the deepest place on Earth, and what we find down there will change everything we think we know about biology, about survival, about the very nature of existence itself. To understand the Mariana Trench, you must first understand the ocean itself, and to understand the ocean, you must reckon with a truth that most of us spend our lives casually ignoring. We live on a water world. That is not a metaphor. That is not poetic embellishment. It is a statement of geological fact. More than 70% of the Earth's surface is covered by ocean. If you could somehow drain every lake, every river, every puddle, every reservoir on every continent, the total volume of that freshwater would represent barely 3% of the water on the planet. The rest, the overwhelming, incomprehensible rest, is saltwater. It fills basins that stretch from pole to pole, basins so vast and so deep that if you took every mountain on every continent and dropped them into the ocean, the water would still cover them with room to spare. We call our planet Earth, we should call it ocean. And yet, for all the millennia that human beings have sailed across its surface, fished its shallows, charted its currents, and written poems about its beauty, we have barely scratched the surface of what lies beneath. By most scientific estimates, more than 80% of the ocean floor remains unmapped, unobserved, and unexplored. We have better maps of the surface of Mars than we do of the bottom of our own seas. We have sent more human beings to the surface of the moon than we have sent to the deepest point of the ocean. The deep sea is, by every meaningful metric, the last great unknown territory on this planet. And at the very bottom of that unknown territory, carved into the floor of the western Pacific Ocean like a wound in the Earth's skin, lies the Mariana Trench. The trench stretches for approximately 1,500 miles in a crescent-shaped arc, curving gently through the Pacific like the sweep of a scythe. It is not wide, relatively speaking. At its broadest point, it spans roughly 43 miles from rim to rim, but what it lacks in width, it more than makes up for in depth. The trench plunges to a maximum depth of approximately 36,000 feet, nearly 7 miles below the surface of the sea. Its deepest point, a narrow slot at the southern end of the trench known as the Challenger Deep, is the single lowest point on the entire surface of the Earth. If you were to take Mount Everest, the tallest mountain on the planet, and drop it into the Challenger Deep, its summit would still be submerged under more than a mile of water. Think about that for a moment. Let it settle in your mind, a mile of water above the peak of the tallest mountain on Earth. That is the scale of what we are dealing with. That is the magnitude of the abyss. The trench exists because of a process called subduction. In the western Pacific, two of the Earth's massive tectonic plates, the Pacific Plate and the smaller Mariana Plate, meet in a slow-motion collision that has been unfolding for tens of millions of years. The Pacific Plate, older, denser, and heavier, is being forced beneath the Mariana Plate, diving down into the Earth's mantle at an angle. As it descends, it drags the ocean floor down with it, creating a deep furrow, a trench, at the point where the two plates converge. This process is not gentle, it is not peaceful. The subduction zone that created the Mariana Trench is one of the most geologically active regions on the planet. Earthquakes rumble through the rock with terrifying regularity. Volcanic activity simmers and occasionally erupts along the arc of islands that traces the trench's western edge. The Earth here is alive in the most literal sense, constantly moving, constantly reshaping itself, constantly reminding us that the ground beneath our feet and beneath our oceans is far less stable than we like to imagine. But for all its geological drama, it is not the rocks and plates that make the Mariana Trench remarkable. It is what lives between them. For most of recorded history, the deep ocean was considered uninhabitable. This was not an unreasonable assumption, given what people knew at the time. The logic was straightforward and on its surface compelling. Life, as understood by the scientists and naturalists of the 18th and 19th centuries, required three things: sunlight, moderate temperatures, and reasonable pressure. The deep ocean offered none of these. Therefore, the deep ocean must be lifeless. It was a syllogism, clean and simple and entirely wrong. The man most often credited with formalizing this belief was Edward Forbes, a British naturalist who in the 1840s proposed what he called the azoic hypothesis. Forbes had conducted extensive dredging operations in the Aegean Sea, dragging nets across the ocean floor at various depths and cataloging the organisms he collected. He noticed a clear pattern: the deeper he dredged, the fewer organisms he found. At depths below about 300 fathoms, roughly 1,800 feet, his nets came up nearly empty. From this observation, Forbes extrapolated a bold conclusion. He declared that life in the ocean ceased entirely below a certain depth. Below 300 fathoms, he argued, the sea was azoic, without life, a dead zone, a biological desert. Forbes was a respected scientist, and his hypothesis was widely accepted. For decades, it shaped the way the scientific community thought about the deep ocean. Why bother exploring a place where nothing lived? Why invest resources and risk lives to probe a barren wasteland? The azoic hypothesis gave the world permission to ignore the deep sea, and for a long time that is exactly what it did. But the azoic hypothesis had a problem. It was based on limited data collected with primitive equipment in a relatively small and unrepresentative body of water. Forbes had dredged the Aegean Sea, a shallow, enclosed basin that is not particularly representative of the open ocean. His nets were crude, his methods imprecise, and his sample size tiny compared to the vastness of the ocean he was attempting to characterize. He had made the classic scientific error of generalizing from insufficient evidence. The first cracks in the azoic hypothesis appeared in the 1860s and 1870s, when improved dredging technology began to pull up living organisms from depths far below Forbes' supposed dead zone. Worms, crustaceans, sea stars, and other invertebrates were hauled up from the ocean floor at depths of 2,000, 3,000, even 4,000 feet. Each new discovery was a nail in the coffin of the azoic hypothesis, but old ideas die hard, and it took decades for the scientific community to fully abandon Forbes' framework. The real revolution came with the HMS Challenger expedition of 1872 to 1876. This was one of the most ambitious scientific voyages ever undertaken, a globe-spanning journey that covered nearly 70,000 nautical miles and lasted more than 3 years. The Challenger was a converted British naval corvette, fitted out with laboratories, dredging equipment, and a small army of scientists. Its mission was nothing less than to survey the world's oceans, measuring depths, temperatures, currents, and the composition of the seafloor, and to collect biological specimens from every depth the expedition could reach. The results were staggering. The Challenger expedition discovered more than 4,700 new species of marine organisms. It mapped the ocean with unprecedented detail, revealing underwater mountain ranges, vast abyssal plains, and deep trenches that had never been charted. And it sounded the deepest point that had ever been measured in any ocean, a spot in the western Pacific that the expedition named with understated British precision, the Challenger Deep. The Challenger expedition did not reach the bottom of the Mariana Trench. Its sounding equipment, essentially a weighted rope lowered to the side of the ship, measured a depth of approximately 26,850 feet in the area that would later be identified as the trench. This was, at the time, the deepest sounding ever recorded, and it sent shockwaves through the scientific community. The ocean was far deeper than anyone had imagined. And the organisms that the Challenger's dredges pulled up from those depths, strange, pale, alien-looking creatures to conditions of crushing pressure and its proved beyond any doubt that Forbes' azoic hypothesis. life existed in the deep ocean. Just a few hardy survivors clinging to existence at margins, but entire communities of organisms, complex ecosystems with their own food webs, their own predators and prey, their own strategies for survival. The deep ocean was not a desert, it was a frontier. But knowing that life exists in the deep ocean and understanding how it exists are two very different things. The conditions at the bottom of the Mariana Trench are so extreme, so removed from what we experience on the surface that strain the imagination. To appreciate a trench, you must first and the physics are, to put it bluntly, letter. The surface of the ocean, the air force of approximately 14 pounds per square of your body. This is one actor. You don't feel adapted to it. Fluids and gases inside your back against the external force, maintaining equilibrium. But as you descend into the water above you, air pressure rapidly. 33 feet increases by atmosphere. A depth easily reachable scuba divers, the pressure 4 atmospheres. 1,000 feet, the submarines operate, it is roughly 31 atmospheres. At the average depth of the ocean, about 12,000 feet, the pressure is approximately 365 atmospheres. And at the bottom of the Challenger Deep, nearly 36,000 feet below the surface, the pressure reaches approximately 1,086 atmospheres. That is nearly 16,000 pounds per square inch. 16,000 pounds pressing against every square inch of anything that ventures down there. To put that in perspective, imagine placing a small coin on a table and then stacking approximately 8 automobiles on top of it. That is the force being exerted on every square inch of every organism, every rock, every grain of sediment at the bottom of the Mariana Trench. At those pressures, the physical properties of matter itself begin to change. Water, which we think of as incompressible, actually does compress slightly at those depths. The water at the bottom of the trench is approximately 5% denser than the water at the surface. Gases are crushed into liquid-like densities. Proteins, the molecular machines that drive every biological process, are deformed by the pressure, their delicate three-dimensional structures warped and distorted. Chemical reactions that proceed at a certain rate on the surface behave differently under those conditions, with some accelerating and others slowing down or ceasing entirely. For any organism evolved for surface conditions, the pressures of the Challenger Deep would be instantly and catastrophically fatal. Cell membranes would rupture, proteins would unfold, enzymes would cease to function, bones would crack and collapse. A human being transported instantaneously to the bottom of the trench would be crushed into something unrecognizable in a fraction of a second. The pressure is not merely hostile to surface life, it is annihilating, and yet life persists. Now consider temperature. Sunlight, the primary source of heat for the ocean's surface, penetrates only the uppermost layer of the water column. The exact depth of light penetration depends on water clarity, angle of incidence, and other factors, but as a general rule, visible light is effectively absent below about 1,000 feet. Below that depth, the ocean is dark, not dim, not twilight, dark, absolutely, completely, profoundly dark, no glimmer, no glow. No faint suggestion of illumination, just blackness stretching in every direction, unbroken and total. Without sunlight, there is no solar heating, and the temperature of the deep ocean drops precipitously. At the bottom of the Mariana Trench, water temperatures hover between about 34 and 39 degrees Fahrenheit, just barely above the freezing point of fresh water. The saltiness of seawater lowers its freezing point to about 28 degrees Fahrenheit, so the water in the trench remains liquid, but only just. It is a cold that seeps into everything, slowing chemical reactions, reducing metabolic rates, and creating an environment where every biological process must operate at a fraction of the speed it would achieve on the warm, sunlit surface. And then there is the darkness itself. We tend to think of darkness as simply the absence of light, as a passive condition, as nothing. But in the Mariana Trench, darkness is not nothing, it is everything. It is the defining characteristic of the environment, the single most important factor shaping the evolution of every organism that lives there. There is no day, there is no night, there is no dawn, no dusk, no season, no variation. There is only darkness, unchanging and eternal, stretching back billions of years to the moment the trench first formed. In a world without light, the rules of biology as we know them on the surface are rewritten from the ground up. Photosynthesis, the process by which plants and algae convert sunlight into chemical energy, the process that forms the foundation of virtually every food web on the surface of the earth, is impossible. Without photosynthesis, there are no plants. Without plants, there is no primary production in the traditional sense. The entire energetic basis of the ecosystem must be found elsewhere, in sources of energy that do not depend on the sun. This is the fundamental challenge of life in the Mariana Trench, not just surviving the pressure, not just enduring the cold, but finding energy, finding food, in a world where the sun's bounty never reaches. How does life solve this problem? The answer is both elegant and startling, and it reveals something profound about the nature of life itself. To understand how the deep ocean feeds itself, you must first understand the concept of marine snow. Marine snow is one of the most important and least appreciated phenomena in the ocean. It is the slow, continuous rain of organic particles that falls from the sunlit surface waters down through the water column toward the ocean floor. These particles are composed of a vast and varied mixture of biological material: dead phytoplankton, the microscopic algae that form the base of the surface ocean's food web; fecal pellets from zooplankton and fish; shed mucus, discarded shells, fragments of dead organisms, bacteria, and other organic detritus. As these particles sink, they aggregate into larger clumps, binding together through a combination of biological and physical processes. The resulting aggregates, sometimes called flocks or marine snow particles, can range in size from tiny specks barely visible to the naked eye to large, fluffy clumps several inches across. From a distance, the steady descent of these particles through the water column looks remarkably like a gentle snowfall, hence the name. But unlike real snow, marine snow is food. It is the primary source of energy for most of the organisms that live in the deep ocean, the thin, fragile thread that connects the sunlit surface to the lightless depths. But here is the catch: marine snow is an incredibly inefficient delivery system. As particles sink through the water column, they are consumed, decomposed, and broken down by bacteria and other organisms at every depth. By the time what remains of the marine snow reaches the bottom of the Mariana Trench, nearly 7 miles below the surface, the vast majority of its organic content has already been consumed. Estimates vary, but most scientists believe that only about 1 to 3% of the organic matter produced at the surface ever reaches the deepest parts of the ocean floor. Some estimates put the figure even lower, at less than 1%. Think about what that means. For every 100 units of food produced by phytoplankton at the sunlit surface, perhaps 1 or 2 units at most make it all the way to the bottom of the trench. The deep ocean is not just dark and cold and under tremendous pressure. It is hungry. It is perpetually, chronically, desperately hungry. This scarcity defines everything about life in the Mariana Trench. It dictates body size, metabolic rate, reproductive strategy, predatory behavior, and virtually every other aspect of the biology of deep-sea organisms. In a world where food is so rare, waste is not an option. Efficiency is not a luxury. It is a requirement for survival, and this leads to one of the most striking characteristics of life in the deep ocean: its patience. Deep-sea organisms are, as a rule, slow. They move slowly, they grow slowly, they reproduce slowly. They age slowly. Their metabolic rates are a fraction of those seen in surface organisms of comparable size. A deep-sea fish might take a decade to reach maturity, where a surface fish of the same size might reach maturity in a year. A deep-sea sponge might live for centuries, growing so slowly that its annual growth rings are barely detectable even under a microscope. Everything in the deep ocean operates on a timescale that would seem glacial to a surface organism. This slowness is not a deficiency. It is a strategy. In an environment where energy is scarce, the organisms that survive are the ones that use the least energy. They are the ones that can slow their engines to a barely perceptible idle, that can wait, motionless and patient, for days or weeks or months between meals, burning as little fuel as possible, conserving every calorie, every molecule of stored energy, against the possibility that food may not come again for a very long time. This is what scientists sometimes call quiet endurance, and it is perhaps the most important survival strategy in the deep ocean. It is not dramatic, it is not flashy, it does not make for exciting television, but it is profoundly effective. The organisms that practice quiet endurance have been doing so for millions of years, surviving in conditions that would kill any surface organism within minutes. Their patience is not passive. It is a finely tuned evolutionary adaptation, honed by eons of natural selection in the most demanding environment on the planet. But the deep ocean is not always quiet, not always patient, not always a slow, measured dance of survival on the knife's edge of starvation. Sometimes food arrives not as a gentle snow of tiny particles, but as a sudden, dramatic bounty. A whale dies at the surface. A massive tuna succumbs to disease or old age. A school of squid is killed by a sudden change in water temperature, and the body, the carcass, begins to sink. This is what marine biologists call a food fall, and it is one of the most important and dramatic events in the ecology of the deep ocean. A whale fall, the sinking of a dead whale to the ocean floor, is the most spectacular example. A large whale can weigh upwards of 100 tons. When it dies and sinks, it carries with it an enormous quantity of organic matter, a bonanza of energy that, to the starving organisms of the deep ocean floor, is the equivalent of a feast descending from heaven. The carcass does not arrive unnoticed. As it sinks, it releases plumes of dissolved organic molecules into the water, chemical signals that spread outward through the darkness like an olfactory beacon. Deep-sea organisms, many of which have evolved extraordinarily sensitive chemical detection abilities, can sense these signals from remarkable distances. Scavengers begin to converge on the carcass long before it reaches the bottom, following the chemical trail through the darkness like wolves following the scent of blood on the wind. By the time the carcass settles on the ocean floor, the first scavengers are already arriving. Hagfish, among the most primitive vertebrates on Earth, are often among the first on the scene. These eel-like creatures, with their jawless mouths lined with rasping teeth, bore into the soft tissue of the carcass and begin feeding from the inside out. They can consume enormous quantities of food relative to their body size, gorging themselves in a frenzy of feeding that seems almost desperate, and in a sense it is. For a hagfish on the floor of the deep ocean, a whale fall may be the most significant feeding event it experiences in years, perhaps in its entire life. The imperative to eat as much as possible, as quickly as possible, is overwhelming. Close behind the hagfish come the amphipods, small crustaceans that swarm over the carcass in enormous numbers. Some species of deep-sea amphipods are specialist scavengers, spending their entire lives drifting through the abyss, waiting for the chemical signal that announces a food fall. When that signal comes, they converge with astonishing speed and in astonishing numbers. Researchers who have placed bait on the ocean floor at abyssal depths have documented swarms of thousands of amphipods appearing within hours, materializing out of the darkness as if conjured by the scent of food. Larger scavengers arrive too. Sea fish, including various species of rattles, eelpouts, and cusk eels, cruise in to feed on the exposed tissue. Crabs and other crustaceans clamber over the carcass, picking at the flesh with their claws. Octopuses, ghostly and pale, glide in from the surrounding darkness, their tentacles probing the carcass for morsels. The initial feeding frenzy, what scientists call the mobile scavenger stage, can last for months. During this time, the carcass is stripped of its soft tissue with remarkable efficiency. The combined efforts of thousands of scavengers can reduce a 100-ton whale to a skeleton in a matter of weeks to months, depending on the depth and the density of the local scavenger population. But the story of a whale fall does not end when the soft tissue is gone. In fact, in many ways, it is only beginning. Once the scavengers have stripped the bones bare, a new community begins to colonize the skeleton. This is the enrichment opportunist stage, during which polychaete worms, small crustaceans, and other invertebrates take up residence in and around the bones, feeding on the organic rich sediment that has accumulated beneath and around the carcass. The decomposition of the whale's tissues has fertilized the surrounding sediment, creating a localized oasis of organic enrichment in an otherwise barren landscape. These opportunistic colonizers can sustain themselves on this enriched sediment for years. And then comes the most remarkable stage of all, the sulfophilic stage, during which bacteria colonize the lipid-rich bones of the whale and begin to break down the fats stored within them. This bacterial decomposition produces hydrogen sulfide, a chemical compound that, while toxic to most surface organisms, serves as an energy source for a specialized community of chemosynthetic bacteria. These bacteria, which derive energy from the oxidation of hydrogen sulfide rather than from sunlight, form the base of a unique food web that can persist on a whale skeleton for decades. This is not merely a feeding event. It is the creation of a temporary ecosystem, a self-sustaining community of organisms that can persist for 50 years or more on the remains of a single whale. Some of the species found at whale falls are found nowhere else on Earth. They are specialists adapted to exploit this particular type of resource, and they spend their lives drifting through the abyss, waiting for the rare and precious signal that announces the arrival of another carcass. The ecology of whale falls illustrates a fundamental truth about life in the deep ocean. Survival is not a continuous process but a punctuated one. Life at these depths is characterized by long periods of scarcity, interrupted by brief, unpredictable episodes of abundance. The organisms that thrive in this environment are those that can endure the scarcity, that can slow their metabolisms to a crawl and wait, sometimes for years, for the next pulse of food. And when that food arrives, they must be ready to exploit it quickly and efficiently, because the feast will not last and the next one may be a very long time coming. This pattern of scarcity and pulses, of patience and opportunism, is one of the defining rhythms of life in the Mariana Trench. It shapes behavior, morphology, and evolution. It produces organisms that are simultaneously among the most patient and the most voracious on Earth, creatures that can wait for years in a state of near suspended animation and then, in an instant, erupt into a frenzy of feeding that would put any surface predator to shame. But marine snow and food falls, important as they are not the only sources of energy in the deep ocean. There is another source, one that does not depend on the sun at all, one that derives its energy not from the biological processes of the surface ocean, but from the geological processes of the Earth itself. Hydrothermal vents. The discovery of hydrothermal vents in 1977 was one of the most significant scientific breakthroughs of the 20th century, a discovery that fundamentally altered our understanding of where and how life can exist. Scientists aboard the research submersible Alvin, exploring the Galapagos Rift in the eastern Pacific Ocean, descended to a depth of approximately 8,000 feet and found something no one had predicted. Superheated water, laden with dissolved minerals and chemicals, gushing from cracks in the ocean floor. The water was shimmering, distorted by temperatures that exceeded 700 degrees Fahrenheit, and it was black with suspended particles of metal sulfides, earning these vents the name black smokers. But the truly astonishing discovery was not the vents themselves. It was the life surrounding them. Clustered around the vents, in water that was a chaotic mixture of near-boiling vent fluid and freezing ambient seawater, were dense communities of organisms unlike anything ever seen before. Giant tube worms, some over 6 feet long, with bright red plumes extending from white tubular casings, stood in dense forests around the vents. Enormous clams, their shells over a foot in length, carpeted the seafloor nearby. Swarms of blind white shrimp jostled and crowded in the shimmering water directly above the vent openings. Crabs, anemones, mussels, snails, and a bewildering variety of other organisms populated every available surface. This was, to put it mildly, not expected. How could such dense, vibrant communities of life exist in the deep ocean, miles from the nearest ray of sunlight, in water laden with chemicals that would be toxic to most surface organisms? The answer, when it came, was revolutionary. The organisms at hydrothermal vents did not depend on photosynthesis. They depended on chemosynthesis. Instead of using sunlight as an energy source to convert carbon dioxide into organic matter, the bacteria at the base of the vent food web used the chemical energy stored in the hydrogen sulfide, methane, and other reduced compounds dissolved in the vent fluid. These chemosynthetic bacteria performed essentially the same function as plants and algae on the surface. They were primary producers, the base of the food web, but they did it without a single photon of light. Their energy came not from the sun but from the Earth itself, from the heat and chemistry of the planet's interior. The implications were profound. For the first time, scientists had discovered an ecosystem that was entirely independent of the sun. The organisms at hydrothermal vents were not relying, even indirectly, on photosynthetic production. They were tapping into a completely separate energy source, one that had been available since the Earth first formed, billions of years before the first photosynthetic organisms evolved. This meant that life did not require sunlight. Life required only energy in any form and a way to harness it. This discovery did not just change our understanding of the deep ocean, it changed our understanding of life itself. The Mariana Trench, with its active geology and its proximity to the subduction zone that gives it its depth, is a prime location for hydrothermal and cold seep activity. While the most famous hydrothermal vent systems are found at mid-ocean ridges, where tectonic plates are pulling apart, vents and seeps also occur in subduction zones, where the intense geological activity associated with one plate diving beneath another creates pathways for heated, chemical-rich fluids to escape from the Earth's crust. In the Mariana Trench and along the Mariana Arc, the chain of volcanic seamounts that traces the boundary between the Pacific and Mariana plates, researchers have discovered active hydrothermal vents and cold seeps that support chemosynthetic communities. These communities are smaller and less dense than the spectacular vent communities found at mid-ocean ridges, but they are no less important. They demonstrate that chemosynthesis is not limited to a few isolated locations, but is a widespread and fundamental process in the deep ocean, one that provides an alternative energetic foundation for life in places where the sun's energy never reaches. Cold seeps, in particular, are important in the trench environment. Unlike hydrothermal vents, which produce superheated water, cold seeps release fluids at or near the ambient temperature of the surrounding seawater. These fluids are rich in methane, hydrogen sulfide, and other reductants, and they support communities of chemosynthetic bacteria that, in turn, support communities of larger organisms. Cold seep communities tend to be more stable and longer-lived than hydrothermal vent communities because the flow of fluid at cold seeps is slower and more persistent. Some cold seep communities have been estimated to persist for thousands of years, creating stable oases of life on the otherwise barren deep-sea floor. The existence of chemosynthesis in the Mariana Trench is a reminder that the energy budget of the deep ocean is more complex than the marine snow from the surface. The deep ocean receives energy from two fundamentally different sources: the biological productivity of the surface, delivered via marine snow and food falls, and the geological energy of the Earth's interior, delivered via hydrothermal vents and cold seeps. These two energy sources interact in complex ways, creating a mosaic of habitats and communities across the deep sea floor, each with its own unique ecology, its own unique species, and its own unique strategies for survival. Now let us talk about the organisms themselves. What does it look like, this life that exists at the bottom of the world? What forms does it take? What strategies does it employ? How has evolution shaped the bodies and behaviors of creatures that live in conditions so extreme that they would kill any surface organism in seconds? The first thing to understand is that the deep ocean, including the Mariana Trench, is dominated not by large, charismatic animals, but by microbes. Bacteria and archaea, single-celled organisms too small to see naked eye, are by far the most abundant life forms in the deep ocean. They inhabit every surface, every sediment, every drop of water. They live in the rocks of the ocean floor, in the walls of vents, in the guts of larger organisms, and in the open water column. They are the foundation of every deep-sea ecosystem, the primary producers and the primary decomposers, the organisms that drive the chemical cycles that make all other life in the deep ocean possible. Of the Mariana Trench of intense scientific for good reason, have evolved to thrive under conditions, and their bio fascinate out of their surface. They're composed of acids that remain flexible and functional at pressures that would solidify membranes of surface organisms. Their enzymes are structured differently, with modifications that allow them to maintain their three-dimensional shape and function at pressures that would denature or unfold the enzymes of surface organisms. These adaptations are not real. They represent fundamental changes at the molecular level, changes that have been shaped by millions of years of evolution under extreme pressure. The study of these pressure-adapted molecules is a field known as piezobiology, from the Greek word for pressure. It is one of the most active and exciting areas of deep-sea. Understanding how extreme pressure has implications not in biology, but for fields as diverse, pharmaceuticals, and materials science. But microbes, fascinating as they are not what captures the public imagination. Think of life in the deep ocean, they think of the animals, the strange creatures that populate forms and begin to belong more than to science. Mariana Trench are among the most characteristics of the trench's deepest. The word hadal comes from underworld, and it refers to the ocean's deepest zone, from about 20,000 feet to the maximum depth. Hadal amphipods are small, shrimp-like creatures, typically a few inches in length, with translucent bodies and large, black eyes. They are scavengers, spending their lives patrolling the trench floor in search of organic matter, and they are astonishingly abundant. When researchers lower baited traps to the bottom of the Mariana Trench, the traps are often swarmed by thousands of amphipods within hours, a testament to both the abundance of these organisms and the intensity of food competition in the deep ocean. The amphipods of the Mariana Trench have attracted particular scientific attention because of their remarkable adaptations to extreme pressure. Their cells contain unusually high concentrations of a molecule called trimethylamine N oxide, or TMAO. This molecule acts as a piezolyte, a substance that counteracts the effects of high pressure on proteins. Under extreme pressure, proteins tend to unfold, losing their three-dimensional structure, and with it, their function. TMAO stabilizes proteins by increasing the amount of water around them, essentially acting as a molecular shield that protects the protein's structure from the distorting effects of pressure. The concentrations of TMAO in Mariana Trench amphipods are the highest ever measured in any organism, and they increase with depth, suggesting a direct relationship between TMAO concentration and pressure tolerance. This adaptation may also set a limit on how deep animal life can exist. Calculations based on the TMAO concentrations found in the deepest living amphipods suggest that at depths beyond approximately 27,000 feet, the concentration of TMAO required to stabilize proteins would be so high that it would begin to interfere with other cellular processes. This may explain why animal life becomes increasingly sparse below this depth, and why the very deepest parts of the trench are populated primarily by microbes, which use different strategies to cope with pressure. Now consider the xenophyophores, among the most bizarre and fascinating organisms in the deep ocean. Xenophyophores are giant single-celled organisms, some reaching several inches in diameter, that live on the deep sea floor. They are classified as foraminifera, a group of protists that construct elaborate shells or tests from materials they collect from their environment. The tests of xenophyophores are typically composed of sediment particles, sponge spicules, and other debris cemented together with organic compounds, creating structures that resemble tiny, intricate sculptures. Xenophyophores are abundant in the Mariana Trench, where they have been documented at depths exceeding 35,000 feet. Their presence at these depths is remarkable because they are not simple organisms. Despite being single-celled, xenophyophores exhibit complex behaviors. They build and maintain their elaborate tests, they forage for food particles on the sediment surface, and they appear to play a significant role in the ecology of the deep sea floor by providing habitat and food for smaller organisms. Their tests create microhabitats, tiny oases of structural complexity on an otherwise featureless expanse of soft sediment, and these microhabitats attract a diverse community of smaller organisms, including nematodes, polychaete worms, and bacteria. The fact that a single-celled organism can achieve such size and complexity at the bottom of the Mariana Trench is a powerful testament to the adaptive potential of life. It challenges our intuitive assumption that extreme environments should produce only the simplest forms of life. Xenophyophores are not simple. They are architecturally sophisticated, ecologically important, and in their own quiet way beautiful. They are a reminder that complexity and extremity are not mutually exclusive, that even in the most demanding environments on Earth, life finds ways to elaborate, to diversify, to create. And then there are the fish. The hadal zone of the Mariana Trench is not teeming with fish. The energy available at those depths is simply too limited to support large populations of vertebrates. But fish do exist there, and the species that have been found are among the most remarkable vertebrates on the planet. The most famous is the Mariana snailfish, Pseudoliparus swirei, described by scientists in 2018 as the deepest living fish ever documented. This small, translucent, tadpole-like fish has been observed at depths exceeding 26,000 feet, gliding slowly over the sediment of the trench floor. It is not a large animal. Adults reach a maximum length of about 11 inches. It is not a fast animal. It moves with a languid, almost dreamlike grace, conserving energy with every slow sweep of its tail. And it is not a beautiful animal, at least not by the conventional standards of surface aesthetics. Its body is pale, almost ghostly, its skin translucent enough to reveal the outlines of its internal organs. Its eyes are small and seemingly non-functional, vestigial remnants of a visual system that in the perpetual darkness of the trench has lost its purpose. But the Mariana snailfish is a miracle of evolutionary engineering. Its body is almost entirely devoid of hard structures. Its skeleton is only partially mineralized, composed more of cartilage than of bone. This is not a deficiency, it is an adaptation. Hard, rigid structures are vulnerable to the crushing pressure of the deep ocean. Under extreme pressure, rigid materials tend to fracture and collapse. By minimizing mineralization, the snailfish has created a body that is flexible and compressible, a body that bends rather than breaks under the enormous pressures of the hadal zone. Its muscles are soft and watery, infused with lipids that provide both energy storage and buoyancy. Its cells contain high concentrations of TMAO, the same pressure-stabilizing molecule found in hadal amphipods. Its metabolism is slow, its energy expenditure minimal. Everything about the Mariana snailfish is optimized for life at the extreme edge of vertebrate existence, a life of minimal energy input, minimal energy output, and maximal patience. The snailfish feeds on the abundant amphipods of the trench floor, cruising slowly through the darkness, sensing the movements and chemical signals of its prey with a lateral line system that is extraordinarily sensitive. The lateral line is a sensory organ found in all fish, a series of specialized receptor cells arranged along the body that detect vibrations and pressure changes in the surrounding water. In surface fish, the lateral line is one sensory system among many, supplementing vision, hearing, and smell. In the Mariana snailfish operating in total darkness, the lateral line is arguably the primary sensory system, the organ through which the fish perceives and navigates its world. This reliance on non-visual senses is a common theme among deep-sea organisms and one that reveals a great deal about the nature of sensory evolution. On the surface, where sunlight is abundant, vision dominates. Eyes are the primary sensory organs of most surface animals, and the visual cortex, the part of the brain devoted to processing visual information, is often the largest and most complex region of the brain. But in the deep ocean, where light is absent, vision becomes irrelevant and other senses take its place. The evolution of non-visual senses in deep-sea organisms is one of the most fascinating chapters in the story of deep-sea biology. In the absence of light, the organisms of the Mariana Trench have developed alternative ways of perceiving their environment that are, in many cases, more sophisticated and more sensitive than the visual systems of surface organisms. Mechanoreception, the ability to detect vibrations and pressure changes in the surrounding medium, is perhaps the most important non-visual sense in the deep ocean. Water is an excellent medium for transmitting vibrations, far better than air, and deep-sea organisms have evolved to exploit this property with remarkable precision. The lateral line system of fish, mentioned earlier, is one example, but mechanoreception is not limited to fish. Many deep-sea invertebrates, including crustaceans, worms, and mollusks, possess specialized sensory structures that detect vibrations in the water, allowing them to sense the movements of predators, prey, and potential mates from considerable distances. In the stillness of the deep ocean, where currents are slow and water movement is minimal, a vibration stands out like a shout in a silent room. The flick of an amphipod's swimming legs, the slow undulation of a polychaete worm, the distant rumble of a shifting sediment slide, all of these produce vibrations that propagate through the water and can be detected by organisms with sufficiently sensitive mechanoreceptors. For an organism living in total darkness, this vibrational information is as rich and informative as a visual landscape is for a surface organism. It reveals the location and movement of other organisms, the topography of the surrounding terrain, and the subtle environmental cues that guide navigation and behavior. Chemoreception, the ability to detect and interpret chemical signals in the environment, is another crucial sense in the deep ocean. Many deep-sea organisms can detect incredibly low concentrations of specific molecules dissolved in the surrounding water, allowing them to locate food sources, identify mates, and sense environmental conditions with extraordinary precision. The chemical plumes released by a food fall, a hydrothermal vent, or a cold seep can spread through the water for miles, carried by the slow, steady currents of the deep ocean, and organisms that can detect these plumes can home in on their sources from great distances. The sensitivity of deep-sea chemoreceptors is genuinely astonishing. Some deep-sea amphipods can detect the presence of food at concentrations so low that they correspond to a few molecules per billion molecules of water. This is roughly equivalent to detecting a single drop of blood in an Olympic-sized swimming pool. The ability to sense such minute concentrations of chemical signals is a direct adaptation to the food-scarce environment of the deep ocean, where the ability to locate and exploit even the smallest sources of nutrition can mean the difference between survival and starvation. But perhaps the most extraordinary sensory adaptation in the deep ocean is not the enhancement of an existing sense but the creation of an entirely new one. And this brings us to one of the most visually spectacular phenomena in the deep sea, bioluminescence. Bioluminescence, the production and emission of light by living organisms, is one of the most widespread and important biological phenomena in the ocean, and it reaches its most spectacular expression in the deep sea. By some estimates, more than 90% of all organisms in the deep ocean are capable of producing light. In the eternal darkness of the abyss, bioluminescence is not a curiosity. It is a way of life. The mechanism is elegant in its simplicity. Bioluminescent organisms produce light through a chemical reaction, typically involving a light-emitting molecule called a luciferin and an enzyme called a luciferase that catalyzes the reaction. When luciferin is oxidized in the presence of luciferase and oxygen, it produces a photon of light. The specific color of the light depends on the chemical structure of the luciferin and the conditions under which the reaction occurs, but in the deep ocean, most bioluminescence is blue or blue-green, corresponding to the wavelengths of light that travel farthest through seawater. The light produced by bioluminescence is what physicists call cold light. Unlike the light produced by incandescent bulbs, fires, or stars, which generates enormous amounts of waste heat, bioluminescent light is produced with remarkable efficiency. Nearly all the energy of the chemical reaction is converted to light, with less than 20% lost as heat. This efficiency is critical in the energy-scarce deep ocean, where every calorie is precious and wasting energy on heat production would be a potentially fatal extravagance. Bioluminescence serves a dizzying variety of functions in the deep ocean, and the ingenuity of these functions is a testament to the power of natural selection operating over millions of years. Some organisms use bioluminescence to attract prey. The most famous example is the anglerfish, that iconic deep-sea predator with its grotesque appearance and its luminous lure. The anglerfish possesses a modified dorsal fin spine that extends forward over its head like a fishing rod, tipped with a bioluminescent organ called an esca. The esca contains bioluminescent bacteria that produce a steady, pulsing glow, and the anglerfish uses this glow to attract curious prey within striking distance of its enormous, gaping jaws. In the darkness of the deep ocean, any source of light is a curiosity, and curious organisms that investigate the anglerfish's lure often find themselves becoming a meal. But the anglerfish is far from the only organism that uses bioluminescence for predation. Many deep-sea squid have bioluminescent organs called photophores scattered across their bodies, and some species can produce rapid flashes of light that startle and disorient their prey, making them easier to catch. Some deep-sea jellyfish trail long, bioluminescent tentacles through the water, creating curtains of light that attract small organisms the way a porch light attracts moths. Other organisms use bioluminescence for defense. When attacked or disturbed, many deep-sea organisms produce sudden, bright flashes of light that serve to startle predators, a response analogous to the flash of a camera in a dark room. The sudden burst of light can disorient a predator for a fraction of a second, providing the prey with a critical window of time in which to escape. Some organisms go further, releasing clouds of bioluminescent material into the water, the deep-sea equivalent of a smoke screen, that confuses and blinds the predator while the prey slips away into the darkness. One of the most sophisticated defensive uses of bioluminescence is counter-illumination. Many deep-sea organisms, particularly fish and squid, have arrays of photophores on their ventral, or lower, surfaces that produce a faint, downward-directed glow. This glow is carefully calibrated to match the intensity and color of the dim light filtering down from the surface, far above. To a predator looking upward from below, the organism's silhouette, which would otherwise appear as a dark shadow against the faintly illuminated water above, is effectively erased. The photophores replace the shadow with light, rendering the organism invisible against the background. It is a cloak of light, a biological stealth technology so effective that it has evolved independently in dozens of different lineages of deep-sea organisms. Counterillumination is a remarkable example of the precision of evolutionary adaptation. The intensity of the photophore light must match the ambient light exactly. Too bright, and the organism would glow conspicuously against the background. Too dim, and the shadow would remain visible. Many counter-illuminating organisms have light-sensing organs on their dorsal surfaces that continuously measure the intensity of the ambient light and adjust the output of their ventral photophores accordingly, creating a dynamic, real-time camouflage system that responds to changes in the environment. Still other organisms use bioluminescence for communication. In a world without light, the ability to produce light on demand is a powerful communication tool. Many deep-sea organisms use species-specific patterns of bioluminescent flashes to identify themselves to potential mates, to signal their presence to members of their own species, and to coordinate behavior. The specific patterns, consisting of flashes of particular durations, intervals, and colors, serve as a kind of luminous language, allowing organisms to exchange information across the darkness. The courtship displays of some deep-sea species involve elaborate sequences of bioluminescent flashes, with males and females exchanging signals in a back-and-forth dialogue that can last for extended periods. The specificity of these signals, each species with its own unique pattern, ensures that organisms can identify members of their own species in an environment where other sensory cues are limited. In the deep ocean, bioluminescence serves the same function that birdsong serves in a forest or bright plumage serves in a meadow. It is the language of attraction, the beacon that draws mates together across the vastness of the dark. Bioluminescence is so pervasive in the deep ocean that, if you were able to descend in a submersible with its lights extinguished, you would not find yourself in total darkness. Instead, you would be surrounded by a continuous shifting display of light, a galaxy of tiny stars and streaks and flashes, each one produced by a living organism, each one serving a specific biological purpose. The deep ocean is not dark in the way we imagine it. It is illuminated, faintly and sporadically, by the collective glow of billions of bioluminescent organisms, a living light show that has been playing, unwitnessed and unappreciated, for hundreds of millions of years. The adaptations of deep-sea organisms are not limited to sensory systems and light production. The physical structures of these organisms, their bodies, their skeletons, their very tissues, have been reshaped by the extreme conditions of the deep ocean in ways that are both subtle and profound. One of the most consistent themes in deep-sea morphology is the reduction or elimination of rigid structures. As we saw with the Mariana snailfish, hard bones and stiff tissues are a liability under extreme pressure. Rigid materials resist compression, and under the enormous pressures of the deep ocean, this resistance can lead to structural failure. Bones crack, shells collapse, rigid cell walls buckle. The organisms that thrive in the deepest parts of the ocean are those that have evolved to be soft, flexible, and compressible, bodies that yield to the pressure rather than resisting it. This principle extends across virtually every group of organisms found in the deep ocean. Deep-sea fish tend to have weakly mineralized bones, if they have bones at all. Many species are predominantly cartilaginous, with skeletons that are more like those of sharks and rays than like those of the bony fish that dominate the surface ocean. Deep-sea crustaceans often have thinner, more flexible exoskeletons than their shallow water relatives. Deep-sea mollusks may have thinner shells or no shells at all. Even the single-celled organisms of the deep ocean have adapted their structures to accommodate extreme pressure, with cell membranes composed of fatty acids that remain fluid and flexible at pressures that would solidify the membranes of surface organisms. This structural flexibility has interesting consequences for body shape and movement. Many deep-sea organisms have a gelatinous consistency, their bodies composed primarily of water with a minimal structural framework. This gelatinous quality gives many deep-sea creatures their characteristic appearance, the translucent, ghostly, seemingly fragile forms that make deep-sea photographs and videos so hauntingly beautiful. Jellyfish, comb jellies, siphonophores, and other gelatinous organisms are abundant in the deep ocean, their soft, water-filled bodies perfectly suited to the high-pressure environment. But the gelatinous quality of deep-sea organisms is not merely a passive accommodation to pressure. It is also an active adaptation to the energy-scarce environment. Gel, composed primarily of water, is metabolically cheap to produce and maintain. A body made of gel requires far less energy than a body made of muscle, bone, and dense tissue. In an environment where every calorie is precious, building a body out of gel is an efficient strategy, allowing organisms to achieve large size without the enormous energy investment that a muscular, heavily skeletized body would require. This is why many deep-sea organisms are surprisingly large for their taxonomic groups. The phenomenon is known as deep-sea gigantism, and it has produced some of the most impressive organisms in the ocean. Giant amphipods, supergiant isopods, enormous squid, and other oversized deep-sea creatures have captured the public imagination for decades. The exact causes of deep-sea gigantism are still debated, but the energy efficiency of gelatinous body plans, combined with the slow growth rates and long lifespans of deep-sea organisms, likely plays a role. An organism that grows slowly but steadily for decades can eventually reach an impressive size, especially if its body plan is metabolically inexpensive to maintain. The way organisms reproduce in the deep ocean is shaped by the same fundamental constraints that shape everything else: scarcity, darkness, and the difficulty of finding a mate in a vast, three-dimensional environment where population densities are extremely low. On the surface, where organisms are abundant and finding a mate is relatively easy, reproductive strategies tend to be diverse and, in some cases, extravagant. Birds build elaborate nests, fish perform intricate courtship dances, insects produce complex chemical signals. These strategies work because the probability of encountering a mate is relatively high, and the cost of investing energy in courtship and display is outweighed by the benefits of successful reproduction. In the deep ocean, the calculus is very different. Population densities are low. The chances of encountering a member of your own species, let alone a member of the opposite sex, at the right time and in the right place, are vanishingly small. In this environment, the luxury of choosiness is a liability. The organisms that succeed are those that can reproduce whenever the opportunity arises, regardless of the circumstances. This imperative has produced some of the most extreme reproductive strategies in the animal kingdom. The most famous and perhaps the most disturbing to human sensibilities is the reproductive strategy of the deep-sea anglerfish. In many species of anglerfish, the males are tiny, just a fraction of the size of the females. A female anglerfish might be a foot or more in length, while a male of the same species might be barely an inch long. The male's body is stripped down to the bare essentials: a pair of large eyes for locating a female, a pair of well-developed nostrils for detecting her chemical signal, and a set of specialized teeth for attachment. When a male anglerfish locates a female, which he does by following her species-specific chemical and bioluminescent signals through the darkness, he bites into her body and holds on. Then something extraordinary happens. The tissues of the male's mouth begin to fuse with the tissues of the female's body. Blood vessels connect, skin merges. The male becomes physically incorporated into the female, a permanent parasitic appendage attached to her body. Over time, the male's body degenerates, his eyes atrophy, his internal organs shrink and disappear. His entire body is reduced to a pair of gonads, testes attached to the female's body that produce sperm on demand whenever she is ready to spawn. The male has in effect given up his existence as an independent organism and become a reproductive organ of the female. This is not parasitism in the usual sense, where one organism benefits at the expense of another. This is a mutual arrangement driven by the extreme difficulty of finding a mate in the deep ocean. By fusing with a female, a male anglerfish ensures that his genes will be passed on. By carrying a permanent source of sperm, the female ensures that she can reproduce whenever conditions are favorable, without having to find a mate each time. It is a bizarre and, to human eyes, grotesque strategy, but it is supremely effective in the context of the deep ocean, where the opportunity to mate may come only once in a lifetime. Other deep-sea organisms have evolved different solutions to the mating problem. Some are hermaphroditic, possessing both male and female reproductive organs, which ensures that any encounter with another member of the same species is a potential mating opportunity. Some reproduce asexually, budding off clones of themselves and eliminating the need for a mate entirely. Some produce enormous quantities of eggs and sperm, releasing them into the water in the hope that at least a few will encounter each other and fertilize in the vastness of the ocean. Each of these strategies is a response to the same fundamental challenge: how to reproduce in an environment where mates are rare, encounters are infrequent, and the margin for error is essentially zero. The diversity of solutions is a testament to the creativity of natural selection and the relentless pressure of reproduction, the most fundamental drive of all living things. To appreciate the full complexity of life in the Mariana Trench, we must also consider the relationships between organisms, the ecological interactions that bind individual species into communities and communities into ecosystems. In the deep ocean, where resources are scarce and survival is precarious, these interactions are often intense, intricate, and surprising. Predation is, of course, a fundamental ecological interaction, and the deep ocean has its share of predators. But predation in the deep ocean looks very different from predation on the surface. On the surface, predators tend to be active, energetic, pursuit-oriented hunters. Lions chase down zebras, hawks dive on rabbits, tuna speed through schools of smaller fish. These predators rely on speed, strength, and aggression to capture their prey. In the deep ocean, where energy is scarce and the cost of active pursuit is prohibitively high, predators tend to be ambush specialists. They wait, they conserve energy. They position themselves in places where prey is likely to pass and then strike quickly when an opportunity presents itself. The anglerfish, with its bioluminescent lure, is a classic example. It does not chase its prey. It attracts its prey, drawing it close with light and then engulfing it with a lightning-fast strike of its enormous jaws. The energy expenditure of this strategy is minimal, a small investment in bioluminescent chemistry and the occasional burst of muscular effort when a strike is made. Other deep-sea predators use even more passive strategies. Some deep-sea spiders, for example, simply sit on the ocean floor, their long legs splayed outward, and wait for small organisms to wander within reach. Some deep-sea worms extend delicate, sticky tentacles into the water and wait for drifting food particles to adhere to them. These organisms are not so much hunting as they are waiting, exercising the patience that is the defining virtue of life in the deep ocean. Competition, too, takes on a different character in the deep ocean. On the surface, where resources are relatively abundant, competition is often a dynamic, aggressive process, organisms fighting for territory, for mates, for food. In the deep ocean, where resources are scarce and organisms are widely dispersed, competition tends to be more indirect, more a matter of efficiency than of aggression. The organisms that succeed are not necessarily the strongest or the most aggressive, but the ones that can extract the most energy from the least food, the ones that can survive the longest on the smallest reserves, the ones that waste the least and conserve the most. And then there is the question that pulses through every aspect of deep-sea ecology, the question that connects the pressure and the cold and the darkness and the scarcity into a single, coherent narrative. How do these organisms find each other? In a world without light, where population densities are low and the distances between individuals can be enormous, the simple act of locating another organism, whether a potential mate, a potential meal, or a potential partner, is a formidable challenge. The solutions that deep-sea organisms have evolved to address this challenge are as diverse and ingenious as the organisms themselves. Chemical signals are perhaps the most common method. Many deep-sea organisms release specific chemical compounds into the water, compounds that serve as identifiers, attractants, or warnings. These chemical signals can persist in the water for long periods and can be detected by other organisms from great distances, providing a kind of long-range communication system that functions in the absence of light. Vibrations and pressure waves are another important method. As we discussed earlier, water is an excellent medium for transmitting vibrations, and many deep-sea organisms can detect and interpret these vibrations with remarkable precision. The movements of a potential mate, the approach of a predator, the settling of a food particle on the sediment surface, all of these events produce vibrations that propagate through the water and can be detected by organisms with sufficiently sensitive mechanoreceptors. And bioluminescence, of course, plays a crucial role. In the darkness of the deep ocean, a flash of light is an unmistakable signal, a beacon that can be seen from considerable distances, and that can convey specific information through variations in color, intensity, timing, and pattern. These three communication channels, chemical, mechanical, and luminous, are the sensory foundation of deep-sea ecology. They replace vision as the primary means by which organisms perceive and interact with their environment, and they have been refined by millions of years of evolution into systems of extraordinary sensitivity and specificity. But there is one more category of ecological interaction in the deep ocean that deserves special attention, because it is arguably the most important of all. It is the interaction that makes much of deep-sea life possible, the interaction that transforms hostile chemistry into usable energy, that converts toxic compounds into nourishment, that turns the raw output of the Earth's interior into the fuel of living systems. Symbiosis. Symbiosis, the intimate, long-term association between two different species, is common throughout the living world. But in the deep ocean, symbiosis takes on a special significance, because it is often the mechanism by which organisms gain access to energy sources that they could not exploit on their own. The most important symbiotic relationships in the deep ocean involve chemosynthetic bacteria. As we discussed earlier, these bacteria derive energy from the oxidation of reduced chemical compounds, hydrogen sulfide, methane, and others, that are released from hydrothermal vents and cold seeps. The bacteria use this energy to produce organic matter from carbon dioxide, just as plants use sunlight to produce organic matter through photosynthesis. But the bacteria do this in the dark, without a single photon of light, tapping into the chemical energy of the Earth itself. Many larger deep-sea organisms have formed symbiotic partnerships with these chemosynthetic bacteria, housing them within their own tissues and providing them with a stable environment in exchange for a share of the organic matter they produce. The giant tube worms of hydrothermal vent communities, perhaps the most iconic organisms of the deep sea, are a classic example. These worms, which can grow to over 6 feet in length, have no mouth, no stomach, and no digestive system. They cannot feed in the conventional sense. Instead, they have a specialized organ called a trophosome, a dense mass of tissue packed with billions of chemosynthetic bacteria. The tube worm absorbs hydrogen sulfide and oxygen from the surrounding water through its bright red plume and transports these chemicals to the trophosome, where the bacteria use them to produce organic matter. The worm then absorbs this organic matter and uses it as food. This is a partnership so intimate that neither partner can survive without the other. The bacteria require the specific chemical environment of the trophosome to function efficiently. The worm requires the organic matter produced by the bacteria to survive. Together, they form a single integrated unit, a composite organism that is more than the sum of its parts. Similar symbiotic relationships are found throughout the deep ocean in a wide variety of organisms. Many species of deep-sea mussels and clams harbor chemosynthetic bacteria in their gill tissues, using the bacteria's metabolic output as a supplementary or even primary food source. Some deep-sea sponges, worms, and other invertebrates have also formed partnerships with chemosynthetic microbes. In each case, the relationship follows the same basic pattern. The larger organism provides the bacteria with a protected environment and access to the chemical substrates they need, and the bacteria provide the larger organism with organic matter that it could not produce on its own. These symbiotic relationships are not static. They are dynamic, responsive, and surprisingly sophisticated. The larger organisms can regulate the activity of their bacterial symbionts, adjusting the flow of chemical substrates and oxygen to match their metabolic needs. Some organisms can even switch between different types of bacterial symbionts depending on the chemical conditions of their environment, maintaining a portfolio of microbial partners that allows them to exploit a range of different energy sources. The ubiquity and importance of symbiosis in the deep ocean underscores a fundamental truth about life in extreme environments. In a world where resources are scarce and the challenges of survival are severe, cooperation is often more effective than competition. The organisms that thrive in the Mariana Trench are not necessarily the strongest, the fastest, or the most aggressive. They are the ones that have found partners, that have formed alliances, that have learned to share the meager bounty of the abyss. Life in the deep ocean is, at its core, a collective enterprise. Let us now pause and consider the broader picture. We have explored the conditions of the Mariana Trench, the crushing pressure, the freezing temperature, the absolute darkness, and the relentless scarcity of food. We have examined the organisms that live there, from the microbes that form the foundation of the ecosystem to the fish, amphipods, and other animals that inhabit the deepest zones. We have marveled at the adaptations that make this life possible, the flexible bodies, the pressure-stabilizing molecules, the bioluminescent displays, and the extraordinary sensory systems that replace vision in a lightless world. But what does all of this mean? What does the existence of life in the Mariana Trench tell us about life in general? The answer, I think, is something profound. For most of human history, our understanding of life has been shaped by our experience on the surface of the Earth. We have defined the requirements of life based on the conditions we know: sunlight, moderate temperatures, reasonable pressure, access to liquid water. We have assumed, implicitly and sometimes explicitly, that life is a delicate, fragile phenomenon, one that requires a narrow range of conditions to exist, and that cannot survive if those conditions deviate too far from the norm. The Mariana Trench challenges this assumption at every level. Life in the trench does not require sunlight. It has found alternative energy sources, chemical energy from the Earth's interior and organic matter raining down from the surface that allow it to thrive in perpetual darkness. Life in the trench does not require moderate temperatures. It has adapted to near-freezing conditions, slowing its metabolism and restructuring its biochemistry to function efficiently at temperatures that would shut down the metabolic processes of most surface organisms. Life in the trench does not require reasonable pressure. It has evolved molecular strategies, including the use of piezolites like TMAO, that allow its proteins and cell membranes to function normally at pressures exceeding 1,000 atmospheres. Life in the trench does not even require abundant food. It has developed strategies for surviving on almost nothing, slowing its engines to a crawl, waiting with infinite patience for the rare pulses of nutrition that sustain it through the long periods of scarcity. What the Mariana Trench tells us in the clearest possible terms is that life is not fragile. Life is tough, life is resilient, life is flexible, life is creative. Given the slightest opportunity, the barest foothold, the most minimal source of energy, life will find a way to exist. And not just to exist, but to diversify, to elaborate, to create complex ecosystems with intricate food webs and sophisticated ecological interactions. This is not a trivial insight. It has implications that extend far beyond the boundaries of marine biology, implications that reach into the deepest questions of science and philosophy. Consider for a moment the question that has haunted humanity since we first looked up at the stars: are we alone? The search for extraterrestrial life is one of the great scientific endeavors of our time. Spacecraft and telescopes probe the cosmos for signs of biological activity on other worlds, scanning the atmospheres of distant exoplanets for chemical signatures that might indicate the presence of living organisms. Robotic probes have explored the surfaces of Mars and the moons of Jupiter and Saturn, searching for evidence of past or present life. For much of the history of this search, the criteria for identifying a habitable world have been based on conditions that are conducive to life as we know it on the surface of the Earth. Scientists looked for worlds with liquid water, moderate temperatures, and exposure to stellar radiation that could drive photosynthesis. These criteria, collectively known as the habitable zone, or more colloquially, the Goldilocks zone, defined a relatively narrow band of conditions within which life was considered possible. But the discovery of life in the Mariana Trench, and especially the discovery of chemosynthetic ecosystems at hydrothermal vents, has dramatically expanded our concept of habitability. If life can thrive in total darkness, at crushing pressures, at near-freezing temperatures, using chemical energy from the Earth's interior rather than sunlight, then the definition of a habitable environment must be much broader than we previously assumed. Consider the moons of the outer solar system. Europa, a moon of Jupiter, has a surface covered in ice, but is believed to have a liquid water ocean beneath its icy crust, an ocean that may be warmed by tidal heating from Jupiter's enormous gravitational pull. Enceladus, a small moon of Saturn, has been observed ejecting plumes of water vapor and organic molecules from cracks in its icy surface, suggesting the presence of a subsurface ocean with active hydrothermal processes. These worlds do not exist in the traditional habitable zone. They are far from the sun, cold, dark, and irradiated. By the old criteria, they would be considered utterly inhospitable to life. But by the new criteria, informed by what we have learned from the Mariana Trench and the deep ocean, they are tantalizing candidates. If life can exist at the bottom of the Earth's ocean, powered by chemical energy from the planet's interior, then there is no reason, in principle, why life could not exist at the bottom of Europa's ocean or Enceladus's ocean, powered by the chemical energy generated by those moons' own geological processes. The Mariana Trench has become, in effect, a laboratory for astrobiology, a place where scientists can study life under conditions that approximate, in some respects, the conditions that might exist on other worlds. The organisms of the trench, with their chemosynthetic metabolisms, their pressure-adapted biochemistry, and their ability to thrive in total darkness, are not just fascinating in their own right. They are models, templates, proofs of concept for the possibility that life exists elsewhere in the universe, perhaps much more widely than we have dared to imagine. This is the deepest lesson of the Mariana Trench, not just that life exists in the abyss, but that the abyss teaches us what life truly is. It teaches us that life is not a fragile candle flame, flickering uncertainly in a hostile universe, requiring just the right conditions to burn. Life is a fire that once kindled is almost impossible to extinguish. It adapts, it evolves, it endures, it finds a way. And if it can find a way at the bottom of the Mariana Trench in the most extreme environment on this planet, then where else might it find a way? The answer, when it comes, may be the most important discovery in the history of science, but we are getting ahead of ourselves. Before we can answer the cosmic question, we must first return to the trench itself, to the organisms and ecosystems that have prompted these grand reflections, and examine them in even greater detail. Because the truth is, for all that we have learned about the Mariana Trench in the past few decades, we have barely scratched the surface of its mysteries. The trench is vast, it is deep, it is difficult and expensive to reach. The technology required to explore it is among the most sophisticated on Earth, and even the most advanced submersibles and remotely operated vehicles can only remain on the bottom for limited periods before the crushing pressure and the corrosive seawater take their toll on equipment. As a result, our knowledge of the trench's biology is fragmentary, based on scattered observations, brief submersible dives, and the analysis of organisms collected by baited traps and sediment samples. For every species we have identified, there are likely dozens, perhaps hundreds, that we have never seen. For every ecological interaction we have documented, there are likely thousands that play out, unseen and unrecorded, in the darkness of the trench. The exploration of the Mariana Trench is an ongoing project, one that will require decades, perhaps centuries, of sustained effort before we can claim anything approaching a comprehensive understanding of its biology. And as we continue to explore, we can be confident that the trench will continue to surprise us, that each new dive, each new sample, each new observation will reveal organisms and interactions that challenge our assumptions and expand our understanding of what life can do. Let us consider in greater depth the microbial world of the trench, because it is here, in the realm of the very small, that some of the most profound adaptations to extreme conditions are found. The sediment at the bottom of the Mariana Trench is not simply mud. It is a complex, living ecosystem in its own right, teeming with microorganisms that have adapted to conditions that would be lethal to virtually any surface organism. Studies of sediment samples collected from the Challenger Deep have revealed diverse communities of bacteria and archaea, representing hundreds of distinct species, many of which are found nowhere else on Earth. These microbial communities are engaged in a complex web of metabolic activities. Some are heterotrophs, organisms that obtain energy by consuming organic matter, breaking down the sparse marine snow and other organic particles that settle on the trench floor. Others are autotrophs, organisms that produce their own organic matter, either through chemosynthesis or through metabolic pathways that are still poorly understood. Still others are lithotrophs, organisms that derive energy from the oxidation of inorganic compounds, such as hydrogen, ammonia, or manganese that are present in the sediment. The metabolic diversity of these microbial communities is staggering. In a single gram of trench sediment, there may be thousands of species of microorganisms, each exploiting a slightly different metabolic niche, each contributing to the overall chemical cycling of the ecosystem. These microbes are not just passive inhabitants of the sediment. They are active agents, driving chemical transformations that affect the composition of the water, the availability of nutrients, and the structure of the sediment itself. One of the most intriguing discoveries about the microbial communities of the Mariana Trench is their unexpectedly high level of metabolic activity. For decades, scientists assumed that the extreme pressure and low temperatures of the Hadal zone would result in extremely slow microbial metabolism. But recent studies have shown that, in some cases, the microbial communities of the trench are actually more metabolically active than those found at shallower depths on the abyssal plain. This counterintuitive finding may be related to the trench's unique shape. The narrow V-shaped profile of the trench acts as a natural funnel, channeling settling organic particles into a relatively small area at the bottom. As a result, the concentration of organic matter in the sediment at the bottom of the trench may be higher than on the surrounding abyssal plain, where organic particles are spread over a much larger area. This concentrated food supply may fuel higher levels of microbial activity, creating a hotspot of biological productivity at the very bottom of the ocean. This funnel effect has important implications for the ecology of the trench. It means that the trench floor, far from being the most barren part of the ocean, may actually be relatively enriched compared to the surrounding abyssal plains. The organisms that live at the bottom of the trench may have access to a more reliable food supply than those that live on the flat, featureless plains at lesser depths. This could help explain the surprisingly high densities of amphipods and other macrofauna that have been observed at the bottom of the trench. The study of pressure-adapted enzymes in trench organisms has opened a window into the fundamental relationship between physics and biology, revealing how the laws of thermodynamics and the mechanics of molecular behavior constrain and shape the evolution of life. Enzymes are the molecular machines that drive every biological process, from the digestion of food to the replication of DNA. They work by binding to specific substrate molecules and catalyzing specific chemical reactions, converting substrates into products with extraordinary speed and precision. The ability of an enzyme to perform its function depends critically on its three-dimensional shape, the precise arrangement of atoms and chemical bonds that creates the active site where the catalytic reaction takes place. Under normal conditions, the shape of an enzyme is determined by the balance between various molecular forces: hydrogen bonds, hydrophobic interactions, ionic bonds, and van der Waals forces. These forces hold the enzyme in its functional configuration, creating the specific three-dimensional architecture that allows it to bind its substrate and catalyze its reaction. But extreme pressure disrupts this balance. Under high pressure, the volume occupied by the enzyme decreases, and the arrangement of water molecules around the enzyme changes. These changes can alter the balance of molecular forces that maintain the enzyme's shape, causing the enzyme to unfold or denature and lose its function. For surface organisms pressure, this denaturation, resulting in the rapid failure of metabolic processes and ultimately death. The organisms of the Mariana Trench enzymes that resist pressure denaturation. These pressure-adapt enzymes achieve their resistance through to their amino acid sequences, arrangement of amino blocks that determines the enzymal structure. The modifications are remarkable, involving the substitution of just a few amino acids at key positions in the protein chain. But all changes have large effects, altering the balance of molecular forces in ways that stabilize the enzyme structure under pressure. Some pressure-adapted enzymes achieve stability by increasing the compactness of their structure, reducing the amount of empty space within the molecule, and making it more resistant to compression. Others achieve stability by modifying the arrangement of hydrogen bonds and hydrophobic interactions in ways that counteract the effects of pressure. Still others rely on the presence of enzymes like TMAO, which, as we discussed earlier, stabilize proteins by altering the behavior of water molecules around them. The study of these pressure-adapted enzymes is not merely an academic exercise. It has practical applications in fields ranging from biotechnology to medicine. Enzymes that function under extreme pressure could be used in industrial processes that operate at high pressures, such as food processing and sterilization. Understanding how pressure affects protein structure could lead to new insights into protein folding diseases, such as Alzheimer's and Parkinson's, in which proteins misfold and lose their function. The Mariana Trench, in this sense, is not just a natural laboratory for studying extreme biology. It is a source of potential innovations, a treasure trove of molecular solutions to problems worked on by evolution for millions of years. As we move through the water column of the trench, ascending from the deepest sediments toward the surface, the character of life changes in ways that are both predictable and surprising. The hadal zone, the deepest zone of the ocean, extends from approximately 20,000 feet to the maximum depth of the trench. This is the realm of the amphipods, the xenophyophores, the snailfish, and the pressure-adapted microbes. Life here is sparse by surface standards, but remarkably diverse given the extreme conditions. The organisms of the hadal zone are specialists, adapted to their specific depth range and often unable to survive at shallower depths, where the lower pressure would disrupt their pressure-adapted biochemistry. Above the hadal zone lies the abyssal zone, extending from approximately 13,000 to 20,000 feet. This is the vast, flat, seemingly featureless expanse of the deep ocean floor, the abyssal plain. Life here is more diverse and more abundant than in the hadal zone, sustained by a slightly higher flux of marine snow and a somewhat lower pressure. Abyssal organisms include a wide variety of sea cucumbers, brittle stars, polychaete worms, isopods, and other invertebrates, as well as various species of deep-sea fish. Higher still, from about 3,300 to 13,000 feet, is the bathyal zone, the region that encompasses the continental slopes and the mid-ocean ridges. This is spectacular deep-sea ecosystems, including the hydrothermal vent communities and the cold seep communities discussed earlier. The bathyal zone is more productive than the abyssal and hadal, receiving a higher flux of organic fish and benefiting from the chemical and geological activity. Relatively abundant with complex fibrate ecological zone from the surface down to 100 feet is the twilight zone of the ocean. This is the transitional light fades from distant world away to the darkness. The mesoplane of the most understood regions of the ocean, volume of water, enormous biome, early in the form squids that undergo migration to the surface and descending to a vertical migration mass movement planet of organisms thousands of feet every day. Mesopelagic zone role in the ocean, transporting from deep via in their own bodies into the rain of it sustains life floor. The layered zone from the darkest trench is of isolated zones, integrated ecosystems and processes of each layer influence and depend upon the organisms and processes of every other layer. The phytoplankton of the surface produce the organic matter that eventually reaches the trench floor. The microbes of the trench floor break down that organic matter and recycle its nutrients back into the water column, where they can, over geological timescales, be returned to the surface by deep ocean circulation. The Mariana Trench is not an isolated pocket of life. It is the deepest node in a global network of biological and chemical processes that connect the sunlit surface to the darkest abyss. Understanding the trench requires understanding this network, and understanding this network requires a perspective that encompasses the entire ocean, from top to bottom, from shore to shore, from pole to pole. We are only beginning to develop that perspective. The technology required to explore the deep ocean is advancing rapidly, driven by innovations in robotics, materials science, sensor technology, and data processing. Autonomous underwater vehicles, remotely operated vehicles, and manned submersibles are reaching depths and durations that would have been unimaginable a few decades ago. New sensors can detect chemical signals, pressure changes, and biological activity with unprecedented sensitivity. Advanced imaging systems can photograph and film deep-sea organisms in their natural habitats, revealing behaviors and interactions that were previously invisible. But for all these advances, the deep ocean remains overwhelmingly unknown. Each expedition to the Mariana Trench brings back data that raises more questions than it answers. New species are discovered on virtually every dive. New metabolic pathways are identified in nearly every sample of trench microbes. New ecological interactions are documented with each deployment of a baited camera or a sediment core. The Mariana Trench is a reminder of how much we still have to learn about our own planet. It is a reminder that for all our technological sophistication, for all our satellite imagery and digital mapping and global communication networks, there are places on Earth that we have barely glimpsed, worlds within our world that remain as mysterious and as alien as any planet orbiting a distant star. And it is a reminder that the most important discoveries are often not the ones that confirm what we already know, but the ones that overturn what we thought we knew, the ones that challenge our assumptions and expand our horizons, the ones that force us to rethink our most basic ideas about what is possible. The Mariana Trench has done this again and again. It has shown us that life does not require sunlight, that life does not require moderate temperatures, that life does not require reasonable pressure, that life requires only energy and the creativity to use it. What else will the trench show us when we look deeper? What other assumptions will it overturn? What other horizons will it expand? We do not know, and that is precisely the point. The deep ocean is the last great unknown frontier on our planet, a place where surprises are not the exception but the rule, where every dive is a journey into genuine uncertainty, where the next observation could change everything. The Mariana Trench is calling, and we are only beginning to listen. We have traveled far in this first part of our journey, from the sunlit surface of the ocean to the crushing depths of the Challenger Deep. We have met organisms that glow in the dark, that survive pressures that would crush steel, that wait for years in patient silence for a meal that may never come. We have explored ecosystems powered not by the sun, but by the chemical energy of the Earth itself. We have glimpsed a world that, until a few decades ago, we did not even know existed, but our journey is far from over. In the second part of this exploration, we will dive deeper still, not in terms of physical depth, but in terms of understanding. We will examine the specific mechanisms of survival in greater detail, exploring the molecular biology of pressure adaptation, the neuroscience of deep-sea sensory systems, and the evolutionary history of the organisms that call the trench home. We will investigate the role of the Mariana Trench in the global ocean system, exploring how the processes of the deep ocean influence the climate, the chemistry, and the biology of the surface world. And we will confront the ultimate question that the trench poses to us. In a universe of billions of stars and billions of planets, is the kind of life we have found in the Mariana Trench a unique miracle, or is it a universal phenomenon repeated countless times on countless worlds across the cosmos? The answers are waiting in the darkness. They have been waiting for billions of years, and we are only now beginning to find them. The abyss is deep, but our curiosity is deeper. This journey continues. We left the darkness still calling. We left the questions still unanswered. We left the trench still waiting as it has waited for millions of years in its silence and its cold and its crushing absolute pressure, indifferent to our curiosity, indifferent to our instruments, indifferent to everything except the slow, patient business of being alive. But we are not indifferent to it. Because the Mariana Trench, as we have begun to discover, is not merely a geographical feature, not merely a crack in the ocean floor, not merely the deepest place on a planet that we have convinced ourselves we understand. It is a mirror. It reflects back to us everything we thought we knew about life, about biology, about the conditions necessary for existence, and it shows us with quiet and irrefutable clarity that we were wrong, that we were thinking too small. That the boundaries we had drawn around the concept of life were not boundaries at all but assumptions, convenient fictions that the trench dissolves the moment we look at it honestly. In the first part of this journey, we explored the physical conditions of the trench, the pressure and the cold and the darkness and the scarcity of food. We met the organisms that inhabit its deepest zones, from the microscopic archaea in the sediment to the ghostly snailfish gliding over the trench floor. We marveled at bioluminescence, at chemosynthesis, at the extraordinary molecular strategies that allow life to function under conditions that should, by every previous understanding, make life impossible. Now we go deeper, not in depth. We are already at the bottom of the world. We go deeper in understanding. We follow the threads of biology and chemistry and evolution further into the fabric of what the trench can teach us. We examine the architecture of survival at the molecular level, the engineering solutions that billions of years of natural selection have produced in this most demanding of classrooms. We explore the role that this hidden world plays in the larger story of the planet, in the cycling of chemicals and carbon and energy that sustains all life on Earth. And we confront head-on the implications of everything we have found for our understanding of life here and for our search for life elsewhere. The darkness is deep, but the story is deeper. Let us begin. To truly appreciate how organisms survive in the Mariana Trench, you need to descend to the molecular level. You need to go smaller than cells, smaller than organelles, all the way down to the level of individual molecules, because it is at this level that the most fundamental adaptations to extreme pressure are found, and it is at this level that the engineering of survival is most clearly revealed. Every living cell is in essence a chemical factory. It takes in raw materials and converts them, through a series of precisely controlled chemical reactions, into the molecules it needs to grow, to reproduce, and to perform its specific functions. These chemical reactions are catalyzed by enzymes, and the accuracy and efficiency of these reactions depend on the precise three-dimensional structures of those enzymes. If the enzymes work correctly, the cell functions. If they do not, the cell dies. The problem with extreme pressure is that it disrupts the delicate balance of molecular forces that maintains the structure of enzymes. We touched on this in the first part of our story, but the implications are worth exploring in more detail because they illuminate something fundamental about the relationship between physics and biology. Consider a protein molecule. Proteins are long chains of amino acids, folded into specific three-dimensional shapes by the complex interplay of chemical forces. Some regions of the protein are hydrophobic, attracted to other hydrophobic regions and repelled by water. These hydrophobic regions tend to cluster together in the interior of the folded protein, away from the surrounding water. Other regions are hydrophilic, attracted to water and comfortable on the exterior of the folded protein. The folding of a protein is, in a sense, a negotiation between these competing forces, a continuous process of adjustment that arrives at the specific three-dimensional configuration that minimizes the total energy of the system. Under normal pressure, this configuration is stable. The forces are in balance and the protein maintains its functional shape, but apply extreme pressure and the balance shifts. Pressure favors configurations that occupy less volume. In a protein, the hydrophobic core, that dense, water-excluding interior, occupies significant volume. Under pressure, the energetic cost of maintaining this hydrophobic core increases, and the protein may begin to unfold, exposing its hydrophobic interior to the surrounding water. This unfolding, called denaturation, destroys the enzyme's three-dimensional structure, and with it, its function. The organisms of the Mariana Trench have evolved two primary strategies to combat pressure-induced denaturation. The first strategy is intrinsic adaptation, modifying the amino acid sequences of proteins to make them inherently more stable under pressure. By substituting specific amino acids at key positions in the protein chain, deep-sea organisms can alter the balance of forces that determine the protein's shape, making it more resistant to the distorting effects of pressure. These substitutions are often subtle, involving just a few amino acids out of hundreds, but their effects on protein stability can be dramatic. Comparative studies of enzymes from deep-sea and shallow water organisms have identified several common patterns in these stabilizing substitutions. Deep-sea proteins tend to have fewer large, flexible amino acid residues in their hydrophobic cores, reducing the compressibility of the core and making it more resistant to pressure-induced denaturation. They also tend to have more compact structures overall, with fewer internal cavities and voids that could collapse under pressure. And they often have modified surface properties that alter how water molecules interact with the protein, reducing the interest of maintaining the protein structure in a high-pressure environment. The second strategy is extrinsic adaptation, using small molecules called piezolites to stabilize protein structure. We discussed TMAO, trimethylamine N oxide, in the first part of our journey, and it deserves closer examination because it is one of the most important and most studied piezolites in the deep ocean. TMAO is a small, simple organic molecule consisting of a nitrogen atom bonded to 3 methyl groups and an oxygen atom. It is found in the tissues of virtually all marine organisms, but its concentration varies dramatically with depth. In shallow water fish, TMAO concentrations are relatively low, typically less than 50 millimoles per kilogram of tissue. In deep-sea fish, the concentration increases with depth. In hadal amphipods from the bottom of the Mariana Trench, TMAO concentrations can exceed 400 millimoles per kilogram, 8 times higher than in their shallow water relatives. How does TMAO stabilize proteins under pressure? The mechanism is subtle and elegant. TMAO does not interact directly with the protein it protects. Instead, it alters the behavior of the water molecules surrounding the protein. Water molecules normally have a certain degree of freedom to move and rearrange themselves around a protein's surface. TMAO, when dissolved in water, restricts this freedom, organizing the water molecules into a more structured arrangement around the protein. This structured water layer stabilizes the protein's surface, making it energetically more costly for the protein to unfold. Mathematically, TMAO shifts the equilibrium between the folded and unfolded forms of a protein toward the folded form. It does not prevent unfolding entirely, but makes unfolding less likely, raising the energetic barrier that the protein must overcome to denature. The higher the pressure, the more TMAO is needed to maintain this equilibrium, which is why TMAO concentrations increase with depth in deep-sea organisms. This relationship between TMAO concentration and depth has been used to estimate the maximum depth at which animal life can exist. As we mentioned in the first part of our journey, calculations based on the highest observed TMAO concentrations in deep-sea organisms suggest that beyond approximately 27,000 feet, the TMAO required to stabilize proteins would interfere with other cellular processes. This physiological limit may set the deepest boundary for animal life, explaining why the very deepest parts of the trench are inhabited primarily by microbes, which use different strategies to cope with pressure. The study of TMAO and other piezolites has opened a new chapter in our understanding of how life adapts to extreme environments. It reveals that adaptation is not always about changing the fundamental machinery of life, about creating entirely new types of molecules with new functions. Sometimes, adaptation is about changing the environment within the cell, about tweaking the chemical context in which the existing machinery operates to make it function correctly under new and challenging conditions. This is a more conservative strategy, one that requires less extensive genetic change, and it may be the reason that adaptation to extreme pressure has occurred independently in so many different lineages of deep-sea organisms. Cell membranes, no less than proteins, present a critical challenge to life under extreme pressure. The cell membrane is the boundary that separates the living cell from its environment. It is a thin, flexible sheet composed primarily of phospholipid molecules, each consisting of a hydrophilic head group and 2 hydrophobic fatty acid tails. These molecules spontaneously arrange themselves into a bilayer, with their hydrophilic heads facing the surrounding water and their hydrophobic tails pointing inward, creating a stable, self-sealing boundary. The function of the cell membrane depends critically on its physical state. At the right temperature and pressure, the membrane is in a fluid state, with individual phospholipid molecules free to move laterally within the bilayer. This fluidity is essential for membrane function. It allows proteins embedded in the membrane to move and interact with each other. It permits the transport of molecules across the membrane. It enables the membrane to deform and reform as the cell changes shape. Extreme pressure, like extreme cold, tends to solidify cell membranes, reducing their fluidity and impairing their function. The effect of pressure on membrane fluidity is similar to the effect of decreasing temperature. Applying pressure to a cell membrane is, in terms of its physical effects, roughly equivalent to cooling the membrane by several degrees. Deep-sea organisms compensate for this pressure-induced solidification through a process called homeoviscous adaptation, a beautiful example of biological feedback regulation. The strategy involves modifying the composition of the fatty acid tails in the phospholipid molecules that make up the membrane. Fatty acids come in 2 main varieties: saturated fatty acids, which have no double bonds between their carbon atoms and pack tightly together, tending to produce a more solid membrane, and unsaturated fatty acids, which have one or more double bonds that create kinks in the carbon chain, preventing tight packing and producing a more fluid membrane. By increasing the proportion of unsaturated fatty acids in their membrane phospholipids, deep-sea organisms counteract the solidifying effect of pressure, maintaining the membrane in an appropriately fluid state even at enormous depths. The deeper the organism, the greater the proportion of highly unsaturated fatty acids in its membranes. This is not a passive response. It is an active regulatory process. Cells monitor the fluidity of their own membranes through specialized sensor proteins and respond to changes in fluidity by adjusting the composition of the fatty acids being synthesized and incorporated into the membrane. The result is a membrane that maintains its optimal physical state across a range of temperatures and pressures, a remarkable example of biological homeostasis operating at the molecular level. The highly unsaturated fatty acids found in the membranes of deep-sea organisms include some that are familiar to nutritionists and health scientists. Omega-3 fatty acids, particularly eicosapentenoic acid and docosahexaenoic acid, compounds that are found in abundance in deep-sea fish and that have been associated with various health benefits in surface organisms. The reason these fatty acids are so abundant in deep-sea organisms is not, of course, that the organisms are trying to be healthy. It is that these specific fatty acids, with their multiple double bonds and their highly kinked structures, are particularly effective at maintaining membrane fluidity under the conditions of extreme pressure and cold that characterize the deep ocean. This is a striking example of how the study of deep-sea biology can yield insights with practical relevance to fields far removed from marine biology. The biochemistry of membrane adaptation in deep-sea organisms has informed our understanding of lipid nutrition, has contributed to the development of dietary supplements, and has provided new perspectives on the relationship between membrane composition and cellular function in health and disease. Deep-sea organisms must also cope with the chemical peculiarities of their environment, and these peculiarities are numerous and often surprising. The water at the bottom of the Mariana Trench is not simply cold, salty seawater. It has a distinct chemical character, shaped by the geological processes of the subduction zone, the slow accumulation of sediment over millions of years, and the biological and chemical transformations carried out by the microbial communities that inhabit it. One of the most important chemical features of the hadal environment is the elevated concentration of certain ions and dissolved compounds relative to the overlying water. The serpentinization of subducted oceanic crust, a process in which water reacts with iron and magnesium-rich rocks to produce serpentine minerals, generates hydrogen gas and hydroxide ions, creating localized environments with unusual chemical conditions. These chemically active fluids can influence the composition of the water throughout the trench, creating gradients and patchworks of chemical conditions that are exploited by different microbial communities. The sediment at the bottom of the trench is a rich archive of chemical information. It is composed not only of organic particles from the surface, but also of volcanic ash from the eruptions along the Mariana Arc, mineral particles derived from the weathering of continental rocks, and the siliceous and calcareous tests of planktonic organisms that have sedimented out of the water column over millions of years. This complex mixture of materials contains a diverse array of chemical compounds, some of which serve as energy sources for chemosynthetic microbes, and some of which are incorporated into the tissues of the organisms that consume those microbes. The chemical environment of the trench also has implications for the carbonate chemistry of the water. At great depths, the high pressure and low temperature favor the dissolution of calcium carbonate, the mineral that makes up the shells and skeletons of many marine organisms. Below a certain depth, known as the carbonate compensation depth, calcium carbonate dissolves faster than it is produced by biological processes, and the sediment becomes depleted in carbonate minerals. The bottom of the Mariana Trench, which lies well below the carbonate compensation depth, calcium carbonate is effectively absent from the sediment. This has important implications for the organisms that live there. Deep-sea organisms that would normally build shells or skeletons from calcium carbonate must find alternative materials or abandon rigid structures entirely, another pressure that drives the evolution of the soft, flexible body plans that characterize the fauna of the hadal zone. The oxygen content of the water in the trench is another critical chemical variable. Oxygen is essential for aerobic respiration, the primary metabolic strategy of most complex organisms. In many regions of the deep ocean, oxygen can be depleted by the bacterial decomposition of organic matter, creating oxygen minimum zones where life must rely on anaerobic metabolism or where it is effectively excluded. In the Mariana Trench, however, oxygen levels in the water column are generally adequate for aerobic life, maintained by the slow circulation of deep water that carries oxygenated surface water to the greatest depths over timescales of centuries. This circulation pattern, part of the global thermohaline circulation that drives the slow overturning of the world's oceans, is critical for maintaining the habitability of the deep ocean. It ensures that even the deepest and most isolated corners of the abyss are connected, however tenuously, to the oxygen-rich surface, sustaining the aerobic metabolism of the organisms that live there. The evolutionary history of the organisms that inhabit the Mariana Trench is a story of profound and sustained isolation, a story of lineages that colonized the deep ocean millions of years ago and have been shaped by its extreme conditions ever since. The question of how organisms came to inhabit the deep ocean is one that evolutionary biologists have been grappling with for decades. The deep ocean is, in many ways, an island, isolated from the surface world by the enormous energetic and physical barriers of the water column. Colonizing it requires not just the ability to survive at great depth, but the ability to get there in the first place, to traverse the hundreds or thousands of feet of increasingly hostile water that separate the surface from the deep sea floor. The colonization of the deep ocean has occurred multiple times, in multiple lineages, over the course of the Earth's biological history. Some deep-sea lineages are extraordinarily ancient, their origins tracing back hundreds of millions of years, to a time when the continents were arranged differently, the ocean basins had different shapes, and the deep ocean may have been chemically and physically different from what it is today. Others are more recent, having colonized the deep ocean within the past few million years, perhaps in response to changes in ocean circulation patterns or the opening of new deep-sea habitats. The process of deep-sea colonization typically begins in shallow water, where an ancestral lineage living at moderate depths gradually extends its range downward, generation by generation, adapting to the increasingly extreme conditions as it goes. The genetic changes required for successful deep colonization are not, in most cases, sudden or dramatic. They are gradual accumulations of small modifications, each one conferring a slight advantage in the deep environment, each one selected for by the relentless pressure of natural selection. But this gradual process of range extension and adaptation is not the only way that deep-sea lineages arise. Some lineages may have colonized the deep ocean via hydrothermal vents or cold seeps, using the chemosynthetic food sources provided by these geological features as a stepping stone to the otherwise food-scarce abyssal and hadal environments. The hypothesis that hydrothermal vent communities have served as refugia, places where deep-sea lineages have survived through geological upheavals that extinguished shallow water populations, is supported by some phylogenetic evidence, though the full story is still being worked out. The isolation of deep-sea populations from each other and from their shallow water relatives has important evolutionary consequences. In the vastness of the deep ocean, barriers to gene flow exist in the form of temperature gradients, pressure gradients, and the simple physical distance between suitable habitats. Populations separated by these barriers can diverge genetically over time, accumulating differences that may eventually result in the formation of new species. This process, called allopatric speciation, is thought to be responsible for much of the biological diversity of the deep ocean. But the deep ocean also presents opportunities for genetic exchange that might not be obvious at first glance. Many deep-sea organisms, particularly the sessile or slow-moving invertebrates of the deep-sea floor, have planktonic larvae juvenile stages that spend a period of time drifting in the water column before settling to the bottom. During this planktonic phase, larvae can be transported by deep ocean currents over enormous distances, potentially connecting populations that would otherwise be isolated. This larval dispersal is thought to be an important mechanism for maintaining genetic connectivity across the vast distances of the deep ocean and for allowing organisms to colonize new habitats as they become available. The genetic diversity of deep-sea organisms is still poorly understood, largely because the tools for studying it, primarily DNA sequencing and genomic analysis, have only recently become accessible for use on organisms collected from the deep ocean. Early studies suggested that many deep-sea species had very low genetic diversity, consistent with small population sizes and limited gene flow. But more recent studies, using more comprehensive genomic methods, have revealed surprising levels of genetic diversity in some deep-sea lineages, suggesting that their populations may be larger and more connected than previously thought. The evolution of sensory systems in deep-sea organisms is a particularly rich area of study, one that illuminates the general principles by which natural selection shapes the organs of perception to match the specific informational environment of a given habitat. We discussed in the first part of our journey how deep-sea organisms have evolved enhanced chemoreception and mechanorception to compensate for the loss of vision in the lightless deep. But the story of sensory evolution in the deep ocean is more complex and more interesting than a simple trade of one sense for others. Consider the evolution of eyes in deep-sea organisms. In the permanent darkness of the hadal zone, functional eyes might seem to offer no advantage. And indeed, some deep-sea organisms, including the Mariana snailfish, have vestigial eyes that appear to be non-functional. These reduced, degenerate eyes are evolutionary relics, structures that were once functional in shallow water ancestors, but have been reduced or eliminated over generations of life in the dark, as the energetic cost of maintaining complex eye tissue outweighed any benefit. But not all deep-sea organisms have degenerate eyes. Many species of deep-sea fish and crustaceans have extraordinarily large, highly developed eyes, organs that are dramatically larger relative to body size than those of their shallow water relatives. These large eyes are not an adaptation to sunlight, they are an adaptation to bioluminescence. In the mesopelagic and bathopelagic zones, where bioluminescence is abundant, the ability to detect the faint flashes and glows of other organisms is a significant advantage. An organism with large, sensitive eyes can detect bioluminescent signals from greater distances, identify the specific patterns of light that indicate a particular species, and track the movements of bioluminescent prey or predators through the darkness. The evolution of large, sensitive eyes in these organisms is not a contradiction of the principle that vision is useless in the deep ocean. It is a demonstration of the principle that evolution always optimizes for the actual informational environment, not for the environment we assume should be present. Some deep-sea fish have taken the evolution of their eyes in remarkable directions. The barrel-eye fish, Macropina microstoma, has eyes that are tubular, barrel-shaped organs oriented upward within a transparent, fluid-filled head. These eyes are extraordinarily sensitive to faint light, and their upward orientation allows the fish to detect the silhouettes of organisms above it, including potential prey that might be visible against the faint downwelling light from the surface. The transparency of the head allows the eyes to rotate forward when the fish needs to look directly ahead, a feature that was not even known to scientists until relatively recently, because specimens captured in nets always had their eyes in the upward pointing position, leading researchers to believe the eyes were fixed. The four-eyed spookfish, a member of the family Opisthoproctidae, has taken eye evolution even further. This remarkable fish has eyes that contain two separate retinas, each receiving light from a different direction. One retina is oriented upward, as in the barrel-eye fish. The other is oriented to the side, using a system of mirrors rather than lenses to focus light. This mirrored eye is a remarkable example of convergent evolution, because mirrors are used in the optical instruments built by engineers precisely because they focus light without the chromatic aberration that affects lenses. The spookfish arrived at this optical solution through natural selection, making it the only vertebrate known to use mirror optics in its visual system. These examples illustrate a broader principle of deep-sea sensory evolution. The absence of sunlight does not eliminate selection pressure for visual acuity. It redirects that pressure toward the detection of bioluminescent light, resulting in visual systems that are optimized for detecting faint, point source signals rather than the broad, diffuse illumination of the surface world. The result is a remarkable diversity of eye designs, each one a creative solution to the problem of seeing in a world lit only by the living things that inhabit it. The acoustic world of the deep ocean is another dimension of sensory experience that deserves careful attention, because sound plays a role in the deep ocean that has few parallels in the surface world. Sound travels remarkably well through water, far better than it travels through air. In air, sound travels at approximately 1,100 feet per second. In seawater, it travels at approximately 5,000 feet per second, nearly 5 times faster. And in the deep ocean, the physical properties of the water create a phenomenon that has important implications for long-range communication. In the ocean, the speed of sound varies with temperature, pressure, and salinity. Near the surface, where the water is warm, sound travels relatively quickly. At greater depths, where the temperature decreases, sound travels more slowly. Below a certain depth, however, the increasing pressure begins to outweigh the decreasing temperature, and sound speed begins to increase again. The depth at which sound speed reaches its minimum is called the SOFAR channel, which stands for sound fixing and ranging. The SOFAR channel acts as a natural acoustic waveguide. Sound produced within the channel tends to be refracted back toward the depth of minimum sound speed, preventing it from escaping to the surface or to the deep sea floor. This means that sounds produced within the SOFAR channel can travel enormous distances without significant loss of energy, propagating through the ocean for hundreds or even thousands of miles. This acoustic phenomenon is not lost on the organisms of the deep ocean. Some species of deep-sea fish produce sounds, low-frequency rumbles, clicks, and pulses that may be used for communication over long distances. Whether these organisms deliberately exploit the SOFAR channel is not entirely clear, but the physics strongly suggest that low-frequency sounds produced at the appropriate depth would be channeled and amplified by the SOFAR effect, extending the communication range of the producing organism far beyond what would otherwise be possible. The deep ocean is not silent. It is filled with sound, the creaking and groaning of tectonic plates, the rumble of distant earthquakes, the calls of whales traveling through the water column, the clicks of shrimp, the low-frequency moans of deep-sea fish. These sounds propagate through the darkness, carrying information about the geological and biological events that produce them to any organism sensitive enough to receive them. Hearing in the deep ocean is therefore not a trivial sense. For organisms that have evolved the ability to detect and interpret the acoustic signals of their environment, sound provides a window onto a world that extends far beyond the limited range of their other senses. An organism in the Mariana Trench can, in principle, detect the acoustic signature of a food fall occurring miles away, or the rumble of a submarine earthquake on the other side of the trench, simply by attending to the vibrations propagating through the water. This long-range acoustic awareness may be a critical component of survival in the deep ocean. For organisms that rely on rare, unpredictable food sources, any information about the location of those sources is enormously valuable. The ability to detect distant acoustic signals may allow deep-sea organisms to home in on food sources that would otherwise be invisible to them, extending their effective foraging range far beyond the limits of their chemical and mechanical senses. The question of how deep-sea organisms find each other for reproduction, which we touched on in the first part of our story, is worth exploring in greater depth because it is one of the most fascinating and poorly understood aspects of deep-sea ecology. In the deep ocean, where population densities are low and the distances between individuals can be vast, the problem of mate finding is severe. An organism that fails to find a mate cannot reproduce, and in a population where every individual's genetic contribution matters, the failure to reproduce is a significant evolutionary cost. Natural selection has therefore placed enormous pressure on the development of effective mate-finding strategies in deep-sea organisms. Chemical communication is the most widespread and seemingly most effective mate-finding strategy in the deep ocean. Many deep-sea organisms, particularly invertebrates such as worms and crustaceans, release specific pheromones, species-specific chemical signals that act as long-range attractants. These pheromones can persist in the water for extended periods and can be carried by currents over considerable distances. Organisms that detect the pheromone of a conspecific, a member of their own species, can follow the chemical gradient upstream to its source, locating a potential mate even in the featureless, lightless expanse of the deep sea floor. The specificity of these chemical signals is remarkable. In a deep sea environment where multiple species may coexist in close proximity, the ability to distinguish the pheromone of your own species from those of other species is critical. The chemical structures of pheromones are therefore highly species-specific, often differing from related species by just a few molecular details, differences that are nevertheless sufficient to ensure that individuals respond only to members of their own species. Some deep-sea organisms have evolved extremely elaborate reproductive behaviors that we are only beginning to document. Deep-sea octopuses, for example, have complex courtship rituals involving visual signals, including bioluminescent displays and chromatophore patterns, that allow individuals to assess the quality of potential mates and make reproductive decisions. How these octopuses navigate the challenges of mate finding in the deep ocean is still not fully understood, but it is clear that they do so effectively, given that deep-sea octopus populations persist and reproduce successfully. The reproductive biology of deep-sea organisms is also shaped by the energetics of the environment. In an energy-scarce environment, the production of eggs and the nurturing of offspring are extremely costly. Deep-sea organisms have responded to this cost in a variety of ways. Some produce very few eggs but invest heavily in each one, providing it with large yolk reserves to sustain the developing embryo through the long period before it can begin feeding independently. Others produce many small eggs with minimal yolk, relying on the planktonic larval phase to exploit surface productivity for growth and development. The choice between these strategies, known as the KR continuum in ecological theory, is mediated by the specific conditions of the organism's environment. In the very deepest parts of the hadal zone, where food is most scarce and the physical challenges most severe, the trend toward fewer, larger, more energetically provisioned eggs is most pronounced. At shallower depths, where conditions are somewhat less extreme, a wider range of reproductive strategies is found. The care of offspring is rare in the deep ocean, simply because the energetic cost of parental care is prohibitive in such a food-scarce environment. Most deep-sea organisms reproduce by releasing their eggs and sperm into the water and leaving fertilization and development to chance. But there are notable exceptions. Some deep-sea fish guard their eggs, and some species of deep-sea octopus are known to brood their eggs, protecting them until they hatch, during which time the female stops eating. This behavior, remarkable in any environment, is extraordinary in the deep ocean, where the energetic cost of brooding, starvation during the brooding period, is potentially fatal. The fact that it occurs at all is a testament to the power of reproductive selection and to the lengths to which natural selection will drive organisms in the service of successful reproduction. The ecological network of the Mariana Trench, the web of feeding relationships, competitive interactions, and mutualistic partnerships that binds its organisms into a community, is far more complex than it might appear at first glance. On the surface, where productivity is high and organisms are abundant, ecological webs are dense and tangled, with many species and many interactions. In the deep ocean, where productivity is low and organisms are sparse, one might expect simpler, more linear food chains. But this expectation is in large part mistaken. The deep ocean, and the Mariana Trench in particular, supports a surprising diversity of ecological roles and interactions. While the total biomass is much lower than in surface ecosystems, the diversity of species, relative to that biomass, can be remarkably high. Some surveys of deep-sea sediment have found hundreds of species in a single core sample, representing a level of biodiversity comparable to that of some tropical surface habitats. This high species diversity in a low productivity environment is one of the great puzzles of deep-sea ecology, and understanding it requires an appreciation of the specific ecological dynamics of the deep ocean. One important factor is the spatial heterogeneity of the deep-sea floor. The trench is not a featureless plain. It has topographic complexity, with ridges, gullies, outcrops of rock, patches of different sediment types, and localized features such as whale falls, hydrothermal seeps, and nodule fields. Each of these microhabitats has its own physical and chemical characteristics and supports a somewhat different community of organisms. The diversity of microhabitats across the trench floor translates into a diversity of ecological niches, which in turn supports a diversity of species. Another important factor is the intermediate disturbance hypothesis, a well-established ecological principle that predicts maximum species diversity at intermediate levels of disturbance. In the deep ocean, disturbances such as turbidity currents, sediment slides, and the sporadic arrival of food falls create a mosaic of habitats at different stages of ecological succession. Areas recently disturbed are colonized by pioneer species. Areas that have been undisturbed for longer periods are colonized by later successional species. The coexistence of multiple patches at different successional stages, each supporting a somewhat different community, contributes to the overall diversity of the ecosystem. The feeding relationships in the trench are also more complex than a simple linear chain from detritus to scavengers to predators. Many organisms are opportunistic omnivores, consuming a wide variety of food sources depending on availability. A hadal amphipod might feed on marine snow particles, on the bacteria coating the sediment surface, on the carcass of a dead fish, or on smaller amphipods, depending on what is available. This dietary flexibility is an important adaptation in an environment where food is unpredictable, allowing organisms to exploit whatever resources are present rather than depending on a single, potentially unreliable food source. The role of microbes in the trench food web is also more complex than a simple base of the food chain position. Microbes are not just producers of organic matter, they are also consumers, decomposers, and transformers of chemical compounds throughout the food web. The microbial loop, a suite of processes by which dissolved organic matter is taken up by bacteria, which are in turn consumed by protists and other microscopic organisms, is an important pathway for energy flow in the deep ocean, channeling organic matter through multiple trophic levels before it is ultimately respired. The role that the Mariana Trench plays in the larger Earth system is one that is often overlooked in discussions of deep-sea biology, but it is immensely important. The trench is not an isolated pocket of life, disconnected from the global ocean and the global climate. It is a node in a planetary system of chemical and biological cycles, a place where processes that began at the surface, billions of cubic miles away in the sunlit upper ocean, are completed in the depths. The biological pump is one of the most important of these global processes. The biological pump is the collective name for the suite of biological processes by which carbon dioxide is removed from the surface ocean and atmosphere and transported to the deep ocean. It begins with photosynthesis, through which phytoplankton in the sunlit surface ocean convert carbon dioxide into organic matter. When these phytoplankton die or are eaten and their consumers produce fecal pellets, the organic matter sinks through the water column, carrying with it the carbon that was originally in atmospheric carbon dioxide. As this organic matter sinks, it is decomposed by bacteria at various depths, returning some of the carbon to the water as dissolved carbon dioxide. But a fraction of the organic matter escapes decomposition and reaches the deep ocean floor, where it is sequestered in the sediment, effectively removing it from the carbon cycle for millions of years. The efficiency of the biological pump, the fraction of surface-produced organic matter that is successfully transported to the deep ocean, varies with location, season, and oceanographic conditions. In regions where the pump is highly efficient, large quantities of carbon are removed from the atmosphere and stored in the deep ocean. In regions where it is less efficient, more carbon remains in the surface ocean and can exchange with the atmosphere. The Mariana Trench, by virtue of its great depth, is one of the most effective carbon sinks on the planet. Organic matter that reaches the trench floor is sequestered at depths so great that it will not be returned to the surface ocean, and thus to the atmosphere, on any timescale relevant to the current geological period. The trench is, in effect, a long-term storage facility for atmospheric carbon, a repository into which carbon removed from the air by phytoplankton is deposited and preserved for millions of years. The microbial communities of the trench play a critical role in this carbon sequestration. By decomposing some of the organic matter that reaches the trench floor and incorporating some of it into their own biomass, these microbes regulate the fraction of organic carbon that is permanently buried in the sediment versus the fraction that is remineralized and returned to the water column. The balance between these two processes determines the long-term effectiveness of the trench as a carbon sink, and it is a balance that is maintained by the metabolic activities of trillions of microbes in the sediment. Understanding the role of deep-sea microbial communities in the global carbon cycle is a research priority because it has direct implications for our understanding of how the ocean responds to changes in atmospheric carbon dioxide. If the deep-sea carbon pump is disrupted, for example, by changes in ocean circulation patterns or by the introduction of pollutants that alter microbial activity, the consequences could propagate through the entire Earth system, affecting the carbon balance of the atmosphere and, ultimately, the climate. The nitrogen cycle in the deep ocean is another planetary scale process in which the Mariana Trench plays an important role. Nitrogen is an essential nutrient for all living things, a key component of proteins, nucleic acids, and many other biological molecules. In the ocean, nitrogen cycles through a complex series of chemical transformations, mediated primarily by microorganisms, that convert nitrogen between its various oxidized and reduced forms. In the deep ocean, the nitrogen cycle is driven primarily by microbial denitrification and nitrogen fixation. Denitrification is the process by which nitrate, the oxidized form of nitrogen, is converted to nitrogen gas, effectively removing nitrogen from the biologically available pool. Nitrogen fixation is the process by which nitrogen gas is converted back to biologically available ammonia, replenishing the pool. The balance between these two processes determines the total amount of biologically available nitrogen in the ocean, and thus the productivity of the surface ocean, where nitrogen is often the limiting nutrient for phytoplankton growth. The deep ocean, including the hadal zone of the Mariana Trench, is an important site of both denitrification and nitrogen fixation. The anoxic or low oxygen conditions in some regions of the deep ocean favor denitrification, while the chemosynthetic and microbial communities of hydrothermal vents and cold seeps carry out nitrogen fixation. The net effect of these processes in the deep ocean on the global nitrogen budget is still an active area of research, but it is clear that the deep ocean is not a passive repository of nitrogen. It is an active participant in the nitrogen cycle, transforming nitrogen compounds and influencing their availability throughout the ocean. The study of the nitrogen cycle in the Mariana Trench has also revealed unexpected complexity in the microbial communities that carry out these transformations. Deep-sea nitrogen cycling microbes include not just bacteria but also archaea, a separate domain of life that was only recognized as distinct from bacteria in the 1970s. Many archaea are adapted to extreme conditions, and they are particularly abundant in the deep ocean, where they may account for a significant fraction of the total microbial biomass. The archaea of the deep ocean include ammonia-oxidizing archaea, which play a crucial role in the nitrification step of the nitrogen cycle, converting ammonia to nitrite and nitrate. The discovery that archaea, rather than bacteria, are the primary ammonia oxidizers in the deep ocean was a significant surprise, one that has required a substantial revision of our understanding of the deep ocean nitrogen cycle. The physical oceanography of the Mariana Trench region is intimately connected to the biology of the trench, shaping the distribution and abundance of organisms through its influence on water chemistry, temperature, and the transport of food and larvae. The deep water that fills the Mariana Trench is part of the global thermohaline circulation, a slow, density-driven overturning of the world's oceans that operates on timescales of hundreds to thousands of years. Cold, dense water formed at high latitudes, particularly in the North Atlantic and the Southern Ocean, sinks to the deep ocean and flows along the bottom in great rivers of cold, dense water called Antarctic bottom water and North Atlantic deep water. These water masses fill the deep basins of the Pacific Ocean, including the Mariana Trench, carrying with them the dissolved oxygen that sustains aerobic life in the abyss. The water in the Mariana Trench is therefore not stagnant. It is slowly but continuously renewed by this global circulation, with residence times in the trench estimated at hundreds of years. The slow renewal of trench water ensures that oxygen levels are maintained above the threshold for aerobic metabolism, even in the very deepest zones where biological oxygen consumption is occurring continuously. The circulation within the trench itself is more complex and less well understood than the large-scale thermohaline circulation. Internal tides, currents generated by the interaction of tidal forces with the complex topography of the trench, are thought to play an important role in mixing water within the trench and in transporting organic particles to the trench floor. Turbidity currents, dense, sediment-laden flows that cascade down the steep walls of the trench and across the trench floor, are another important transport mechanism, delivering pulses of organic-rich sediment to the deep trench environment. These physical processes interact with the biological processes of the trench in complex ways. Internal tides may resuspend organic particles from the trench floor, making them available to filter feeding organisms in the water column. Turbidity currents may bury organisms under sudden deposits of sediment, creating localized disturbances that reshape the community structure of the trench floor. The interaction between physical oceanography and biology in the trench is a frontier of research, one that requires the integration of physical and biological observations and models to fully understand. Human exploration of the Mariana Trench has a history that is both brief and dramatic, a story of technological ambition, scientific discovery, and the irresistible pull of the unknown. The first descent to the Challenger Deep, the deepest point of the Mariana Trench, was made on January 23, 1960, by Swiss engineer Jacques Piccard and United States Navy Lieutenant Don Walsh aboard the bathyscaphe Trieste. The descent took approximately 5 hours, during which the Trieste withstood pressures that no human-occupied vessel had ever experienced. When the Trieste settled on the bottom of the Challenger Deep at a depth of approximately 35,800 feet, Pickard and Walsh became the first human beings in history to reach the deepest point on Earth. What they saw there was, in its own quiet way, revolutionary. They reported seeing a flatfish resting on the sediment, an observation that, if accurate, demonstrated conclusively that vertebrate life could exist at the very deepest point of the ocean. Their dive lasted only 20 minutes on the bottom, limited by the duration of life support and by the need to return to the surface before nightfall, but in those 20 minutes they established a fact that changed our understanding of the limits of life. The deepest place on Earth was not empty. For more than 50 years after the Trieste's historic dive, no human being returned to the Challenger deep. The extreme depth and the extraordinary engineering challenges of reaching it meant that subsequent exploration was conducted using unmanned vehicles, remotely operated systems, and acoustic instruments. These instruments revealed a great deal about the trench, but they could not provide the direct, immersive observation that a human presence would allow. Then in March of 2012, filmmaker and explorer James Cameron descended to the Challenger deep in a specially designed submersible, the Deep-Sea Challenger, becoming only the third human being in history to reach that depth and the first to do so solo. Cameron spent approximately 3 hours on the bottom, conducting scientific observations, collecting samples, and filming the trench in high definition. His observations confirmed and extended what earlier surveys had suggested. The Challenger deep is not barren. It is inhabited by microbes, by small invertebrates, by the characteristic organisms of the Hadal Zone, all going about their quiet business in the darkness of the deepest place on Earth. Cameron's dive and the scientific data collected during it sparked renewed interest in Hadal exploration and contributed to a significant expansion of Hadal research programs around the world. New remotely operated vehicles and landers, specially designed to withstand the pressures of the Hadal Zone, have been deployed in the trench in increasing numbers over the past decade, collecting video footage, biological samples, and environmental data from depths that were previously inaccessible. The results have been revelatory. Each expedition to the Challenger deep has returned with new species, new observations, and new insights into the ecology and biology of the Hadal Zone. The Mariana snailfish, now recognized as the deepest living fish ever documented, was discovered during one of these expeditions. New species of amphipods, polychaete worms, and other invertebrates have been described. The microbial diversity of the trench sediment has been characterized using genomic methods, revealing a complex and highly adapted community of microorganisms that is unlike anything found in shallower environments. Perhaps most significantly, these expeditions have revealed something unexpected about the human presence in even the most remote environments. Chemical analysis of amphipods collected from the Mariana Trench has detected the presence of persistent organic pollutants, including polychlorinated biphenyls and polybrominated diphenyl ethers, at concentrations comparable to or higher than those found in organisms from the most heavily polluted coastal environments. These compounds, the products of industrial activity, have found their way to the very deepest point of the ocean, carried there by the same process that carries food, the rain of organic particles from the surface. The discovery of industrial pollutants in the Mariana Trench is a sobering reminder that no place on Earth, not even the most remote and inaccessible, is truly beyond the reach of human activity. The chemicals produced by our industries, released into rivers and coastal waters, enter the ocean and are incorporated into organic particles that eventually sink to the bottom of the sea. The same biological and physical processes that deliver food to the organisms of the trench also deliver our chemical legacy, concentrating it in the bodies of organisms that have never seen the sun and that have no connection to the surface world except through the thin thread of marine snow. This finding has implications both for the conservation of the deep ocean and for our understanding of the global reach of human environmental impact. The Mariana Trench, long regarded as too remote to be affected by human activity, is not immune. And if the deepest point of the ocean is contaminated, then every other part of the ocean is contaminated too. The future of Mariana Trench research is, by any measure, extraordinarily exciting. Advances in autonomous underwater vehicle technology are opening up new possibilities for long-duration, high-resolution exploration of the deep ocean. Current remotely operated vehicles require a surface ship and a tether connecting the vehicle to the ship, limiting their range and endurance. New generations of autonomous underwater vehicles, untethered and self-guided, can navigate the complex terrain of the trench floor independently, following pre-programmed routes or making real-time navigation decisions based on sensor data. These vehicles can remain underwater for days or weeks, covering vast areas and collecting continuous streams of environmental data. Advances in sensor technology are also transforming what is possible in deep-sea research. New chemical sensors can detect and quantify dissolved compounds in the water with unprecedented sensitivity and specificity, allowing researchers to map the chemical environment of the trench in real time. New acoustic imaging systems can create detailed three-dimensional maps of the trench floor at resolutions that were previously unachievable. New genomic sampling devices can collect biological material and preserve it at high pressure until it can be returned to the surface for analysis, overcoming one of the major challenges of deep-sea microbiology, the fact that many organisms die or change their biochemical composition when brought to the surface under decompression. The application of genomics and metagenomics to deep-sea research is perhaps the most transformative development of the past decade. Metagenomics, the direct sequencing of all the genetic material in an environmental sample, allows researchers to characterize the microbial communities of the deep ocean without the need to culture individual organisms in the laboratory. This is a significant advantage because most deep-sea microorganisms cannot be cultured under laboratory conditions. They require the specific physical and chemical conditions of their natural environment to survive and grow, conditions that are extremely difficult to replicate in a laboratory setting. Metagenomics bypasses this limitation, providing a comprehensive census of the microbial community from a single environmental sample. Metagenomic studies of Mariana Trench sediment have already revealed a level of microbial diversity that far exceeds what could have been detected by culture-based methods. Hundreds of novel microbial lineages, representing organisms with metabolic capabilities that have never been described before, have been identified in trench sediment samples. The functional capabilities of these organisms, the specific biochemical reactions they can perform, are being inferred from their genomic sequences, opening a window into the metabolism of organisms that have never been observed directly. Among the most intriguing findings from these metagenomic studies is evidence for novel metabolic pathways, biochemical reactions that do not match anything in the existing catalog of microbial metabolism. Some of these novel pathways appear to be involved in the degradation of complex organic compounds that were previously thought to be resistant to microbial breakdown. Others appear to involve the coupling of unexpected electron donors and acceptors in ways that generate chemical energy from reactions that were not previously recognized as biologically useful. These discoveries suggest that the deep ocean is not just a repository of familiar biology operating under unusual conditions. It is a crucible of metabolic innovation, a place where the extreme conditions and the scarcity of conventional energy sources have driven the evolution of entirely new ways of making a living. The implications of deep-sea research for the search for extraterrestrial life have never been more tangible or more exciting than they are today. The convergence of advances in deep-sea exploration, exoplanet astronomy, and the study of solar system bodies with potential subsurface oceans has created a unique moment in the history of astrobiology. We know from the study of the Mariana Trench and other deep-sea environments that life requires only energy and liquid water. We know that it can exploit chemical energy from geological sources when solar energy is unavailable. We know that it can function under pressures exceeding 1,000 atmospheres at temperatures approaching the freezing point of water in complete darkness with minimal nutrient supply. We know that it can be extraordinarily patient, waiting for decades between significant feeding events. And we know that, given these minimal conditions, it will diversify, elaborate, and create complex ecosystems. Now consider Europa. Europa is a moon of Jupiter, approximately the size of Earth's moon, with a surface covered in a relatively young layer of water ice. The surface of Europa is crisscrossed by a complex network of cracks, ridges, and chaotic terrain that strongly suggests the existence of a global liquid water ocean beneath the ice. This subsurface ocean is thought to be kept liquid by tidal heating, the flexing of Europa's interior by Jupiter's enormous gravitational pull, which generates heat that prevents the ocean from freezing. The depth of Europa's subsurface ocean is uncertain, but estimates range from 10 to 100 miles. If the ocean is approximately 100 miles deep, it contains more liquid water than all of Earth's oceans combined. Pressure at the base of such an ocean would be immense, comparable to or exceeding the pressures at the bottom of the Mariana Trench. More importantly, there is evidence that Europa's ocean floor may have hydrothermal activity. The tidal heating that keeps the ocean liquid also flexes and fractures the rocky seafloor, creating pathways for the exchange of chemicals between the rocky interior and the liquid ocean. If this tectonic activity is accompanied by hydrothermal vent systems, as is suspected, then Europa's ocean floor could have the chemical energy sources, the hydrogen, hydrogen sulfide, and other reduced compounds that support chemosynthetic life on Earth. The analogy with the Mariana Trench is not perfect. Europa's ocean is isolated from the vacuum of space by a layer of ice of uncertain thickness. The chemical composition of its ocean is unknown. Its geological activity may be fundamentally different from that of Earth, but the basic conditions, liquid water, a source of chemical energy, and the basic ingredients of organic chemistry appear to be present. In Enceladus, Saturn presents an even more compelling case. Enceladus actively vents water vapor, ice particles, and organic molecules from its south polar region through a series of cracks known as tiger stripes. The material being vented has been analyzed by the Cassini spacecraft, which flew through the plumes on multiple occasions. The analysis revealed the presence of water vapor, carbon dioxide, methane, molecular hydrogen, and complex organic molecules, all the ingredients, in other words, for chemosynthetic life. The molecular hydrogen is particularly significant. Molecular hydrogen is produced on Earth by the reaction of water with iron and magnesium-rich rocks in a process called serpentinization, the same process that occurs in subduction zones and along mid-ocean ridges. On Earth, this hydrogen serves as an energy source for chemosynthetic microbes. The presence of molecular hydrogen in the Enceladus plumes suggests that serpentinization and possibly hydrothermal activity is occurring at the bottom of Enceladus's subsurface ocean. If that is the case, then the bottom of Enceladus's ocean may be remarkably similar, in its essential features, to the environment around a hydrothermal vent on the floor of the Mariana Trench. And if life can exist around such vents on Earth, then the question of whether life exists in Enceladus's ocean becomes one of the most urgent and most exciting questions in contemporary science. The answer is not yet known. The technology required to penetrate Enceladus's ice layer and directly sample its ocean does not yet exist, though several mission concepts have been proposed and studied. But the question is no longer merely speculative. It is grounded in specific, detailed knowledge of the conditions that life requires, knowledge derived in large part from the study of the deep ocean and the Mariana Trench. There is a philosophical dimension to the study of the Mariana Trench that deserves consideration, a dimension that goes beyond the specific findings of deep-sea biology and touches on something fundamental about the human relationship with the unknown. For most of human history, our relationship with the deep ocean has been defined by ignorance and fear. The deep sea was the realm of monsters, of sea serpents and krakens, of cosmic terrors lurking beneath the waves. It was a place to be avoided, a void that swallowed ships and men with indifferent malice. Even as we have grown more scientifically sophisticated, the deep ocean has retained an aura of menace, of hostile otherness that colors our perception of it. The discoveries of the past few decades have not eliminated this sense of otherness. The organisms of the Mariana Trench are genuinely alien, genuinely strange, genuinely outside the bounds of ordinary human experience. The anglerfish with its parasitic males, the tube worm with its bacterial symbionts, the xenophyophore spreading its single-celled body across the sediment in elaborate architectural patterns, these are not creatures that fit easily into any familiar category, but the discoveries have fundamentally changed the emotional valence of that strangeness. The deep ocean is no longer a void, it is a world. Not a world hostile to life, but a world shaped by life, animated by life, sustained by the creativity and resilience of living things that have been adapting to its conditions for hundreds of millions of years. It is not a place of cosmic horror, but a place of cosmic wonder. This shift in perception matters. It matters for science, because organisms that are seen as wonderful are more likely to be studied than organisms that are seen as terrifying. It matters for conservation, because environments that are appreciated are more likely to be protected than environments that are feared. And it matters for our broader cultural relationship with the natural world, because how we perceive the deep ocean reflects and shapes how we perceive nature in general, as something to be conquered and exploited, or as something to be understood and respected. The Mariana Trench teaches us, if we are willing to learn, that life is not our exclusive possession, not something that exists only where we have placed it or where we find it convenient. Life is a property of the universe, expressed wherever the minimal conditions for its existence are met, in the sunlit surface ocean and in the crushing darkness of the abyss, in the volcanic heat of hydrothermal vents and in the near-freezing water of the trench floor. This is not a comfortable lesson. It challenges our sense of uniqueness, our feeling that life, at least complex and diverse life, is something special, something rare, something that needs our protection and our stewardship because it is so easily destroyed. But the Mariana Trench suggests that life is not easily destroyed, that it is, in fact, very difficult to destroy. That once established in a suitable environment, life persists through geological upheavals, mass extinctions, and the slow grinding of tectonic plates, adapting and evolving and finding new ways to endure. This does not mean that we can afford to be complacent about the conservation of the deep ocean. The discovery of industrial pollutants in the bodies of hadal amphipods is a clear demonstration that human activity can reach even the most remote and seemingly inaccessible environments. The deep ocean is vulnerable, in ways that we are only beginning to understand, to the cumulative effects of pollution, climate change, and ocean acidification. The organisms of the Mariana Trench may be extraordinarily resilient in evolutionary terms, but they are not immune to the chemical and physical disruptions that human activity is imposing on the global ocean system. The lesson is not that we need not worry about the deep ocean because life there is so tough. The lesson is that we should be humbled by the tenacity of life in the trench, inspired by its creativity, and motivated by that inspiration to ensure that we do not become the agents of its destruction. As the biological and physical data from the Mariana Trench accumulates, patterns are beginning to emerge that were not visible in the early, fragmentary studies. One of the most striking of these patterns is the extraordinary degree to which the trench's biology is shaped by its geology, and vice versa. The subduction of the Pacific Plate beneath the Mariana Plate is not just the geological process that created the trench. It is an ongoing process that continuously shapes the trench's environment, influencing the chemistry of the water, the structure of the sediment, and the distribution of energy sources that sustain the trench's ecosystems. Earthquakes along the subduction zone periodically trigger turbidity currents that sweep across the trench floor, resuspending sediment, redistributing organic matter, and creating localized disturbances that alter community structure. Slow, steady serpentinization of the subducted oceanic crust releases hydrogen that diffuses upward into the trench water, potentially serving as an energy source for hydrogen-oxidizing microbes. The biological processes of the trench, in turn, influence its geology. The burrowing and bioturbation activities of infaunal organisms, the worms, crustaceans, and other invertebrates that live in the sediment, mix and disturb the sediment, influencing its physical and chemical properties. The metabolic activities of microbial communities drive chemical transformations that alter the composition of the porewater in the sediment and the overlying water column. The accumulation of organic carbon in the trench sediment, and its eventual burial and transformation, is part of the long-term geological record of the ocean's biological productivity. This two-way interaction between biology and geology is a theme that runs through all of Earth science, and that is particularly well illustrated in the extreme environment of the Mariana Trench. The trench is not just a geological feature that happens to contain some biology. It is a geological-biological system in which the living and the non-living are so thoroughly intertwined that distinguishing them is, in many cases, an artificial exercise. This insight that the boundaries between the biological and the geological are less sharp than we tend to assume is one of the deepest lessons that the Mariana Trench has to offer. It echoes the concept of the Gaia hypothesis, the idea that the Earth functions as a single self-regulating system in which living organisms and physical processes are coupled in ways that maintain the conditions necessary for life. Whether one accepts the full Gaia hypothesis or not, the study of the Mariana Trench strongly supports the view that life is not merely a passenger on a geological Earth, but an active participant in the Earth's chemical and physical evolution. The silence of the trench is not the silence of emptiness. It is the silence of patience, the silence of endurance, the silence of life operating on timescales that dwarf the span of human civilization. The organisms of the Mariana Trench are the products of hundreds of millions of years of evolution, shaped by conditions that have remained in their essential features remarkably consistent over geological time. The pressure of the deep ocean has not changed significantly since the trench formed. The darkness has been unbroken since long before the first complex organisms evolved. The scarcity of food, the cold, the chemical peculiarities of the water, These have been constants, not variables, in the evolutionary history of the trench's inhabitants. This evolutionary stability has produced organisms that are exquisitely, precisely adapted to their environment. They are not approximations, not compromises, not organisms that are making the best of a bad situation. They are perfectly suited to the conditions of the trench, their bodies and biochemistries honed by eons of selection to function optimally in conditions that would destroy any less adapted organism instantly. But this very perfection of adaptation carries a risk. Organisms that are perfectly adapted to a specific set of conditions may be poorly equipped to cope with changes in those conditions. The organisms of the Mariana Trench have evolved in an environment that has been, by surface standards, extraordinarily stable. They have not been exposed to the fluctuations of temperature, chemistry, and food availability that have driven the rapid adaptive evolution of surface organisms. As a result, they may have limited capacity to respond to rapid environmental change. This is a concern because the deep ocean is changing. Not as rapidly as the surface ocean, where temperature increases and acidification driven by the absorption of carbon dioxide are already measurable, but changing nonetheless. The gradual warming of the deep ocean, driven by the penetration of warming signals from the surface over timescales of decades to centuries, is altering the temperature structure of the water column. Changes in ocean circulation patterns, driven by the melting of polar ice and the disruption of thermohaline circulation, are altering the delivery of oxygen and organic matter to the deep ocean. The accumulation of pollutants in the deep sea food web is introducing chemical stressors that have no natural precedent in the evolutionary history of deep sea organisms. How the organisms of the Mariana Trench will respond to these changes is unknown. The timescales on which we can observe deep sea communities are too short, and our baseline knowledge of the trench's biology is too limited to make confident predictions. What we can say is that the trench's organisms have survived far greater upheavals than anything we are currently imposing on them. Mass extinctions, ice ages, profound changes in ocean chemistry, all of these have come and gone while the organisms of the deep ocean have endured. But those past upheavals unfolded on geological timescales. The changes we are currently imposing on the ocean are unfolding on human timescales, decades rather than millions of years. The question is whether evolution, which requires many generations to produce adaptive change, can keep pace with change that is occurring at unprecedented rates. It is a question without a comfortable answer. And it is a question that makes the study of the Mariana Trench and the organisms that inhabit it more urgent than ever. Let us, in these final passages of our journey, return to where we began. Let us return to the darkness. Picture it again, the absolute absence of light, stretching in every direction, up and down and sideways, unbroken and total. The crushing weight of 7 miles of water above you. The cold that permeates everything, slowing every reaction, quieting every movement. The silence that is not the absence of sound, but the presence of a sound too low and too constant for our ears to register. The barely perceptible groan of the earth's crust under the weight of the ocean above. And in this darkness, this cold, this silence, life. A xenophyophore spreads its single-celled body across the sediment, constructing its intricate test of sediment particles and sponge spicules with a precision and care that seems impossibly architectural. An amphipod glides through the water column, its chemoreceptors alert to the faintest trace of organic compounds dissolved in the water, its large dark eyes scanning for any glimmer of bioluminescence that might indicate the presence of prey or competitor. A snailfish cruises slowly along the trench floor, its lateral line vibrating with the subtle pressure changes produced by the movements of the amphipods it hunts, its slow, even movements a study in energy conservation, in the art of achieving maximum effect with minimum expenditure. Far above, nearly invisible through the immensity of water, a speck of organic matter drifts downward. A dead diatom, a fecal pellet, a fragment of mucus, marine snow, food. The amphipod detects it first, a trace of dissolved organic compound carried down in the particle's microscopic wake. It adjusts its course, rising toward the source, its swimming legs beating in the precise, economical rhythm of an organism that has never learned to waste a calorie. In the bacteria that coat the sediment surface, metabolic reactions that would be familiar to a biochemist proceed at a fraction of their surface world speed, but proceed nonetheless, breaking complex organic molecules into simpler compounds, releasing energy, constructing new molecular machinery, replicating DNA, doing all the things that living things do, slowly, patiently, enduringly. And in a crack in the trench wall, where water seeping through fractured rock carries traces of hydrogen sulfide upward into the water column, A community of chemosynthetic bacteria builds organic matter from carbon dioxide and chemical energy, independent of the sun, independent of the surface, independent of everything except the rock and the water and the ancient heat of the Earth's interior. Around them, tiny invertebrates graze on the bacterial mats. Slightly larger organisms graze on the tiny invertebrates, a food web small but complete, sustained by the Earth itself. This is the Mariana Trench. This is the life it contains. And this life, quiet and patient and extraordinarily adapted as it is, tells us something of incalculable importance. It tells us that life is not a fragile anomaly in a universe of dead matter. It tells us that life is a robust, persistent, creative force that will find expression wherever the minimal conditions for its existence are present. It tells us that our definition of minimal conditions has been far too narrow, shaped by the limited range of environments we happen to inhabit and observe. It tells us that the universe of possible life is far larger, far more diverse, far more strange and beautiful than we have yet imagined. And it reminds us that we have barely begun to explore it. The Mariana Trench has been waiting for 7 miles beneath the surface of the Pacific Ocean for tens of millions of years. It has been accumulating secrets, sheltering organisms, hosting ecological dramas of remarkable subtlety and complexity, all in total darkness and silence, unwitnessed and unrecorded. We have known of its existence for less than 200 years. We have explored it directly for less than 60. We have brought back enough data to fill scientific journals and challenge textbooks, but not enough to claim anything approaching complete understanding. Each answer we have found has revealed 3 new questions. Each organism we have described has hinted at dozens more waiting to be discovered. Each metabolic pathway we have characterized has suggested entirely new categories of biological possibility. The Mariana Trench is not a mystery solved. It is a mystery deepened, a frontier that retreats before us as we advance, revealing new complexity, new strangeness, new wonder at every step. This is as it should be. The universe should be larger than our understanding of it. Nature should be more creative than our theories. Life should be more resilient, more inventive, more pervasive than our models predict. The history of science is a history of discovering that the world is bigger, stranger, and more interesting than we thought, and the Mariana Trench is among the most compelling chapters in that history. We are drawn to explore it, not just because it is scientifically important, though it is. Not just because it contains resources that might be economically valuable, though it may. Not just because it holds clues to the origin and evolution of life on Earth and potentially on other worlds, though it does. We are drawn to explore it because it is there, because it is unknown, because the human impulse to look into the darkness and ask what is there is one of the most fundamental and most admirable characteristics of our species. The trench will continue to yield its secrets slowly, reluctantly, one dive at a time, one sample at a time, one genomic sequence at a time. Each piece of knowledge we extract from it will be hard-won, requiring sophisticated technology, careful analysis, and the patient, dedicated work of scientists who have devoted their careers to understanding one of the most challenging environments on the planet. But the knowledge is worth it, because what the Mariana Trench is teaching us at the deepest level is not just about biology or geology or chemistry. It is teaching us about possibility. It is expanding the horizon of what we know to be achievable, of what we know to be real, of what we know to be alive. And in a universe where we are still struggling to understand our own place, where we are still asking the oldest and most fundamental questions about the nature of life and the prospects of its existence beyond our own world, that expansion of possibility is perhaps the most valuable thing of all. The quiet depths are still calling. Every time a research vessel drops a lander over the side and watches it disappear into the blue-black water below, the trench receives it with the same patient indifference that it has shown to everything. It does not care about our curiosity. It does not care about our technology. It does not care about our questions or our theories or our desire to understand it, but it answers us anyway. Not willingly, not eagerly, but inevitably, because the universe does not withhold its secrets from those who look carefully and persistently enough. The trench answers us with organisms we have never imagined, with biochemical strategies we have never conceived, with ecological relationships we have never anticipated, and with the quiet, irrefutable demonstration that life is more resilient, more inventive, and more widely distributed than anything our previous experience prepared us to believe. This is the gift of the abyss. Not the resources it might contain, not the species it might yield for pharmaceutical research, not even the specific answers it provides to the question of extraterrestrial habitability. The gift is larger than any of these things. The gift is perspective. The gift is humility. The gift of understanding that the light we live in, the moderate temperatures we enjoy, the comfortable pressures we never think about, the abundant food we take for granted, these are not the prerequisites of life. They are merely the conditions under which we happen to have evolved. Life itself is older, tougher, and far more resourceful than any particular set of conditions that it happens to currently occupy. The organisms of the Mariana Trench have been alive in various forms for hundreds of millions of years. They survived the mass extinction that killed the dinosaurs. They survived the ice ages that covered the continents in glaciers. They survived the changes in ocean chemistry that accompanied the great oxygenation events of the Precambrian. They have endured quietly and patiently through upheavals that reshaped the surface of the earth, while maintaining their slow, careful, energy-efficient existence at the bottom of the world. They will, in all probability, outlast us. But while we are here, while we have the curiosity and the technology and the desire to look into the darkness and ask what is there, we owe it to ourselves and to the life we have discovered there to look carefully, to look completely, to look with the full capacity of our scientific and creative intelligence, bringing to bear every tool we have developed, every theory we have constructed, every insight we have won from the slow, difficult, magnificent work of science. The Mariana Trench is the deepest place on Earth. It is not the end of our exploration. It is the beginning of our understanding. The darkness is deep, but we are learning to see within it, and what we see there changes everything. There is a place on this planet where the sun has never reached, a place where the pressure would crush steel, where the cold seeps into everything, where silence is not an absence but a presence. It is not on some distant world orbiting a faraway star. It is here. It is beneath us. It is at the